We are delighted to invite our distinguished resource person, Dr. Sivakumar Garu, IASR, onto the dais. We call upon our honorable guest speaker for today, Dr. Kishori Garu from Hyderabad, Pharma, onto the dais. With great respect, I request our distinguished keynote speaker, Professor K. Nagmohan Garu, onto the dais. Now it gives my immense pleasure and privilege to invite our honorable chief guest, Dr. Y.V. Nancharaya Garu, senior scientist, BAR, Chennai, onto the dais. Now I request all the dignitaries on the stage to join lamp lightning ceremony. Thank you so much, sir. Now I call upon stage biotechnology second year students to perform a prayer song.
Stand up.
जय जय हे भगवती सुरभारती तव चरणों प्रणमामः। जय जय हे भगवती सुरभारती तव चरणों प्रणमामः। नाद ब्रह्ममयी जय वागेश्वरी नाद ब्रह्ममयी जय वागेश्वरी शरणं ते गच्छामः शरणं ते गच्छामः। जय जय हे भगवती सुरभारती तव चरणं प्रणमामः। ललितकलामयी ज्ञानविभामयी ललितकलामयी ज्ञानविभामयी वीणा पुस्तकधारिणी वीणा पुस्तकधारिणी। मतिरास्थानो तव पदकमले मतिरास्थानो तव पदकमले ऐ कण्ठा विषहारिणी ऐ कण्ठा विषहारिणी। जय जय हे भगवती सुरभारती तव चरणं प्रणमामः तव चरणं प्रणमामः।
Fantastic group job, most devotional. Now I request C.H. Sirisha Madam to deliver contact sitting about this program.
Good
Morning, everyone. The theme of this conference, Trailblazing Pathways in Life Sciences, highlights the new directions and opportunities in the field of biology. Life sciences today are helping us to address some of the world's biggest challenges like improving human health, ensuring food security, protecting our environmental and advancing sustainability. Current research is opening new doors from breakthroughs in cancer biology that offer hope for better treatments to bioremediation technologies that use microbes and plants to clean up polluted environment. These are just a few examples of how life sciences are shaping a healthier and more sustainable future. Through this conference, we aim to explore such innovations, share research findings, and create a platform where scientists, teachers, and students can learn from one another. We believe these discussions will inspire new collaborations and future discoveries. Thank you.
Thank you so much, madam, for giving a brief intro about today's program, the Trailblazing Pathways in Life Sciences. Now we have opening remarks by our college principal, Dr. M. Ramesh Garu.
Good morning to all of you. Respected guests on the dais, Dr. Y.V. Nancharya Garu, Professor Nagamohan Garu, Dr. Kishore J.R.K., and Dr. Sivakumar V., and other dignitaries on the dais. And my dear students. So, the departments of Biological Sciences is organizing a two-day national conference on trailblazing pathways in life sciences. These biological sciences or chemical or physical sciences, whatever they are, there are a lot of changes in the sciences. What you are studying is totally different from that of what is happening in the real society. There is a lot of advancement in the each and every science. It may be medical sciences or chemistry or physics or biological sciences, each and every area, there are a lot of advancements. You should understand the changes that are taking place in your areas, in your subjects, and unless you study all those things, unless you understand all those things, you cannot go to the higher education in those areas. Our conventional subjects is only a base for us, and advanced subjects you should learn a lot. Then only you can get the good opportunities in your life. So you can use the library for understanding of the changes in your subjects in a better way, or also you can use your mobiles. All of you are using mobile phones. You can use your mobile phones also to understand the changes that are taking place in your subjects. So make use of all those things in a positive way instead of using them for other purposes. So if you want to settle the life in a better way, if you want to settle in a way in which you like, you have to study all those things in a better way. You should understand all those things in a better way. Then only you can settle or you can have the life what you have, what you want to like, remember. So on this occasion, I would like to congratulate the departments of biological sciences for arranging of this type of wonderful conference, two-day conference, and they invited most eminent personalities from different areas. You can enjoy a lot. You can understand the changes that are taking place in the sciences. Thank you for giving this opportunity.
Thank you so much, sir, for your valuable message and brief introduction about today's conference. Now I request D. Shravani Madam to introduce about chief guest.
Good morning, everyone. Myself D. Shravani, faculty in botany, PT Siddhartha College of Arts and Science. Today, I feel euphoric to present the profile of chief guest, that is nothing but Dr. Y.V. Nancharaya from Baba Atomic Research Center. Sir awarded Masters from Andhra University, and he is a PhD candidate from Kalpakam. He is leading as head biofouling and biofilm process section. Head at present, he works on role of microbial communities and sustainable development. His work on role of microbial communities and sustainable development, which is an enormous concern towards environment and sonorous, mentioning about the microbial community work, skills of Nancharaya Gayaru is biofilm making, microbiology in environment application, wastewater treatment, usually applying biotechnology in our environment also. He is also senior researcher with Marie Curie Fellowship. He is also a guest scientist in investigation of plasmid transfer to biofilm bacterial community in bioaugmentation experiments, and many more achievements backed by sir. Thank you for giving me the opportunity.
Thank you so much, madam, for briefing the chief guest profile in a very short view. Now I request our chief guest, Y.V. Nancharaya Garu, doctorate, to give a message about this two-day conference.
Thank you, madam, for the introduction. A very good morning to all the respected dignitaries on the dais and the faculty members and students. It is truly an honor for me to be here as part of this event, which is organized by BB Siddhartha College of Arts and Science. And this coming to this place actually reminds me about my past. I mean, small interaction with Siddhartha College. Way back in 1993, when we completed our MScs in biochemistry at Andhra University, there was a request from PB Siddhartha, this institution, to recommend a faculty for starting a biochemistry course in undergrad, BSc. And fortunately, I was asked by our gurus at biochemistry department that are you willing to go? However, I could not take up because my interest was taking up the research, PhD and things like that. But it was chosen by one of my classmates and a good friend of mine, and who came here and he worked for 2 years. Yeah. And then now I came to know through my another friend, Dr. Shivakumar, that we actually wrote MSc entrance examination in this campus. Okay. So it is like back then, no, actually I had little knowledge about Siddhartha group. In fact, I used to see VR Siddhartha Engineering College in the Poranki when I traveled to Machilipatnam, always. But today I came to know, yesterday and today, that the group is so big, it has a large number of institutions and its umbrella. And one of the premier educational research group based at Vijayawada. And this reminds me back to what is done in Coimbatore, because I live in Chennai, Tamil Nadu. And Coimbatore, there is a group called PS, Govindasamy, a group of institutions. And they have built world-class, top-notch institutions I'm referring only to the private institutions, not to the government. Yeah. So the PSG, they started their institutions way back in before independence, that is 1920s. And they have, you name it, any private institution, medical, engineering or sciences, they are the top. They have a top-notch, top-class medical institution, similarly engineering. In fact, now they have institutions exclusively for R&D. So they have Institute of Advanced Studies where I actually be part of research advisory board. So I relate something like what is happening in Coimbatore as PhD group to Siddhartha group at Vijayawada in different fields of education, particularly giving high quality education from let us say KG to PG or even advanced, okay, including medicine. So I said that I was interested in the PhD program after MSc. Then I was like, I was thinking that because I was thinking that is the only option available for life science candidates at that point of time. Because of, under the impression that there were no there were no job opportunities. So I thought the PhD option, I will get my degree. At the same time, I also get a fellowship for the living purpose. So then I started looking into the options and interacting with various institutions at that time. Based on the journals we read, scientific journals, no? I came across, I was visiting various institutes in India. Also, I wrote to one of the institutes, that's called BRC, Baba Atomic Research Center, one of the scientists, who is an editorial member of one of the journals. And then I wrote him saying that those days there was no email and no even Googling, etc. So we have to literally send a letter by post. Then fortunately he has replied me with a large yellow color with an employment news advertisement indicating there is a job opportunity for science postgraduates, including life sciences, physics, chemistry in BS. So he wrote me a letter saying that instead of PhD opportunity, in fact he denied giving PhD opportunity, but instead he suggested me apply for a job position. So you apply for it and then you have a position, once you get into it, you have an option to execute your PhD. While doing working in BRC, you can also do PhD. So that was his suggestion. As per his advice, I applied for that. I was successfully cleared the written test followed by the entrance examination, written test followed by the interview. Both were conducted at Mumbai. So that was my first visit to Mumbai, let us say. So I was also hesitant to go there as a non-speaking Hindi. My Hindi was very limited, being from a rural background. But I said, okay, let me go because they are ready to give to and fro after second class. So after going there, the first day written test was there, second day there was the interview, and they declared, they did not announce the result, but they gave me their expenses, train expenses. I came back again to Andhra University. Within a week, they sent me a fax saying that you are provisionally selected for the post of scientific officer. You may submit your documents for the verification. So that's how I actually entered Baba Atomic Research Center, and then I just crossed 30 years of service there. So why I want to mention this is that sometimes we overlook some things may not come to our notice. Let's say an opportunity of job employment. Incidentally, I wrote to somebody and then he has suggested me, that's how I entered the place. Okay? But we should be ready for the opportunity. That is what I would like to say here. And coming to the conference part, before coming to the conference part, DRC, there is always a Baba Atomic, there is an atom in it, name itself. So Baba is the one Homi Baba who founded the institution during Nehru time. And then this institution is meant for actually doing nuclear research or building nuclear power reactors. But it is not just that. The institution has entered into many top class research program because they wanted to be like a multidisciplinary research institute, R&D institution. So we have a field from life sciences, then physical sciences, chemistry, then of course engineering, because a large number of engineers work related to the development of technology for reactors, nuclear power reactors. We have about, I think, 25 power reactors across the country now. But apart from that, there's a lot of R&D is going on. Some of the fields, just to mention, like life sciences, they have a microbiology, and there is a research going on utilizing this radiation for creating different varieties of plants with high yield varieties, including groundnut, rice, etc. Also, there are like research. Also there is a cancer care, which is also done from a department of atomic energy. So irradiation, radiation therapy that is being used for treating the tumors. And not just that, there is also molecular biology, then agriculture, then food technology. Radiation is also used for sterilization. If you work in a microbiology lab, petri dishes that is commonly used, which are actually irradiated, radiation sterilized. That's what is done at US. Okay. So coming to my own work, I work in the field of microbiology. So we have there are two areas we work on. One of the work that is going on is on these infectious diseases. In nature, these microbes, when we say a bacteria, we need a microscope to view them. But typically in the nature, as we live as communities, like satellite city, etc., microbes also live like a communities. They don't live in isolation. They come together and grow as a communities in nature. But there are some disadvantages, advantages of these communities. So we call them, let's say, bad microbes or bad microbial communities in the case of health, because they are related to causing that disease. But at the same time, these microbial communities are helping natural cleanup of environment, water, etc. So there is again biotechnology is playing a role. So environmental biotechnology, environmental engineering, etc. So the conference, today conference, the title was actually interesting to me because the title, the main interest is to look into the advances or recent trends that is happening in the field of life sciences, innovations, let us say. And we have a few experts who are coming from different fields, from different premier institutions to deliver the talks. And of course, the students will get benefited by that. And coming to, there are many inventions that have happened actually in the, just to name some of the inventions in the field of life sciences that have impacted the society. I'm not going into the details of those, but just to mention, let's say PCR, RT-PCR, that has been widely used in the case of COVID-2 for detection of SARS-CoV-2. And there are like DNA sequencing and then amplification sequencing, then rDNA technology, which is used for providing transgenic plants. Also, then IVF also is successfully used in in vitro fertilization, and antibiotics, vaccines. So, but these are like big discoveries that have come into the society, but there are many things that are behind these many scientists, and a lot of work has gone into different fields. So similarly, environmental protection is important when we develop. So society develops or human develops, society has to be taken care because resources have to be preserved for the future generations. Because we come and go, but the next generation needs all these resources, be it water or good quality air or soil. So that's where sustainable development is key, I think. And there also again microbiology or genetic engineering or plants play a big role, maintaining the cleanup of the environment. So yeah. So in my talk I will be giving some of the aspects related to the environment, but there are other speakers who will deal about give their expertise in the fields of medicine or advanced life sciences fields. So with this, I would like to thank, I would like to extend the heartfelt congratulations to the organizing team and also the management for inviting me here. And I wish all the students and the faculty to have a pleasant experience during the today conference. Thank you.
Thank you so much, sir, for your valuable message. Sir has given a lot of information about his personal experience and as well as microbial techniques in advance. Sir has given from very basic level of microbial techniques to the advanced level, how they are up to the mark of how the PCR and RT-PCR is going to be happen. And without any further delay, we would like to move on a very special part of today's inaugural session, the launch of our conference, Savanil. So this Savanil captures an essence of our theme, that is trailblazing pathways in life sciences. So I already mentioned that think ahead. So we are always in that line of thinking ahead. So this was given by our director, sir. And sir has motivating not only students, all the faculty members too, for having such a wonderful conference today. And I request all the department faculty members to so this will be revealed by our chief guest. All the department members should accompany on the stage. Degrees and the faculty members. I request all the faculty members of various departments, computer science, management, chemistry, and Telugu department, commerce department, to please join us at the stage, please.
For information I am giving, and nearly about 100 papers and we received.
Thank you so much, sir. Thank you all the departments for that. Thank you so much, sir, for participating in that inaugural launch of Savanil. And now I request Professor P. Srinivas, sir, Head of the Department of Botany, to deliver oath of science.
Good morning to one and all. Thank you, such a prayer that cannot be seen and touched. It must be felt by heart. So thank you one and all. I feel honored and privileged to get the opportunity to propose vote of thanks on this special occasion. I would like to thank all the honorable delegates who blessed us with their presence here. I'm also very much thankful to all the invited speakers and guests. Words are not enough to thank their support and encouragement to present here. On behalf of organizing committee, convey my deep regards and heartfelt thanks to Honorable Director Sir, Sri Vemuri Babu Rao garu. I'm also thankful to our Principal Garu, Dr. M. Ramesh Garu, and Dean Dr. Rajesh C. Jampala Garu, Vice Principal Dr. Manoranjani Madam Garu. They helped us in obtaining necessary administrative approval for organizing the event in this campus. They supported in all possible manner to organize this conference in successful way. My special thanks to our chief patrons of this conference, Sri M. Rajay Garu, President, Siddhartha Academy of General and Technical Education, Sri P. Lakshman Rao Garu, Secretary, Sri S. Venkateswarlu Garu, Treasurer, Sri N. Lalita Prasad Garu, Convener PBSCAC, and Sri Ramesh Garu. So I wholeheartedly thankful to our management team. My sincere thanks to all our faculty colleagues and also non-teaching staff members who always stand by us and motivate us. I feel proud and thank you to each one of you for making this event a special and successful way. I'm very much thank all other faculty members in this college and the students who participate in this conference. I would like to thank and mention into this our Canara Bank people to supporting in one way to other way in financially. At the end, I would like to take this opportunity to thank to all of you for making this event successful. And I wish to enjoy this conference. Thank you, Vanessa.
So one more I have to mention, and all this is because of Professor S. Saraji Garu, and who worked here, and most of them, and maybe they are colleagues for him, and he is a professor in Oman, and only because of him, and we could arrange all these, we can arrange this program. And special thanks to him. And at the same time, we got nearly about 10 papers and from computer science, and 5 papers from Department of Artificial Intelligence and Data Science, and 1 paper and we got and from English department, and 1 from Statistics department also. So that is how, and even the students, faculty members, and they are also involved in this. And special thanks to all of them for making it a grand success. Thank you once again.
Thank you so much, sir. With this, we have done our inaugural session and we have technical sessions. Session 1 with by our chief guest. So the technical sessions begins now. I invite the chairperson for today's first technical session 1, Dr. M. Manoranjani Madam, Head of the Department of Chemistry, to occupy the stage. So first technical session will be started by our chief guest, Dr. Y.V. Nansharay Garu. Sir is going to talk the first session, and rapporteur for this session is D. Shravani Madam. I invite our chief guest, Dr. Y.V. Nansharay Garu, to occupy the stage for first session.
Today our first session will be on role of microbial communities in sustainable development. And this topic will be delivered by Dr. Y.V. Nansharay. And I deem it as an honor and privilege to read out the profile of Dr. Y.V. Nansharay, PhD, FBRS, FASCH, Scientific Officer H. Baba Atomic Research Center, and Professor Life Sciences Homi Baba, National Institute, Mumbai, Maharashtra. Sir has already explained to us that he has successfully completed 30 years full-time in Baba Atomic Research Center. At present, he is the head Biofouling and Biofilm Process Section, IG CAR, and he has been heading the department since July 2021. And he has been a scientific officer H since 2019, and his skills are bioremediation, environmental microbiology, wastewater treatment, environmental biotechnology, marine biology, environmental science, ecotoxicology. And he has been working as a scientific officer C during the year September 1995. And the same topics, he has been researching on the same topics. And during the year 2014-15, he was awarded the prestigious Marie Curie Fellow Award as a senior researcher in UNESCO IHE Institute for Water Education. And the topic of research during that time was environmental biotechnology. Later on, he was a postdoctoral researcher during the tenure November 2010 to February 2011 in Arizona State University Center for Environmental Biotechnology as a full-time postdoctoral researcher, USA, Phoenix, Arizona, United States. The topic is extracellular electron transfer in microbial biofilms. And during the year October 2009 to February 2010, he worked as a postdoctoral researcher in Brookhaven National Laboratory, Mount Upton, New York, United States. The topic was electron shuttles and ionic liquids in Clostridium fermentative metabolism. And also as a guest scientist from August 2001 to June 2002, TU Munchen, and as a scientific trainee in Bath during the year August 1994 and July 1995. He did his MSc from Andhra University in the field of biochemistry and also PhD in life sciences Bath. Sir, we are very much privileged to have you, sir, amidst us today. Now over to our chief guest, Dr. Nancharaya. Thank you.
Madam, for the introduction. Yeah. Very good morning to all of you. I again thank the organizers for giving me this opportunity to present what we are doing at BRC Kalpa. Microbiology, actually microbiology, so you have any, I think, botany, zoology, and then all those traditional subjects, but later on it is evolved into microbiology, biochemistry, and then biotechnology is the latest trend. In my opinion, we need fundamental knowledge of all basic biology disciplines to do a good biotechnology or to apply life sciences information in the form of a technology. Okay. So I'm going to give a brief of how microbes live in nature and what problems they cause us, and how we make them, how message can go into the, you can acquire some knowledge from here. So traditionally, microbes are grown like individual cultures in the lab. Okay, when we go to the lab, what culture you are working? I'm working with Pseudomonas. I'm working with the E. coli. So it is like that, and they are either grown in the form of in the aerobic conditions or anaerobic conditions. So you can see before growth and after growth, right? It is a turbid. That is means there is a growth of microorganism under the anaerobic conditions. Anaerobic, aerobic refers to differences. We are aerobes. We need oxygen for living, right? So similarly, bacteria live under different growth conditions. There are microbes. They require oxygen. They require oxygen for thriving. So for doubling and then for doing metabolism and all that. There are microbes. They strictly grow under completely anaerobe. That means no oxygen should be there. There are microbes which grow in between, facultative. Okay. There are microbes which use nitrate instead of oxygen. They use nitrate for their respiration. Okay. So this is the image, microscope image showing individual cells, let's say, or a bunch of cells. Okay. So this is how the laboratory growth is done. But in the field, they grow like this. So there is like a mat, and you can see that it is very slippery also. Okay. So anywhere if you find some fountain, the surface will be slippery. That's because there is a microbial growth over there, and these microbes produce sticky biopolymers. So even in the bathroom, there is a stagnant water. You should be careful putting your step. So you can see that the surface will be slippery after 2 or 3 days. That's because there are microbes in the water, and they go and attach to the surface. They attach and grow on the surface. During the process, they produce polymers that make them give this slippery, or we call them a slime also. Okay. And if you see under a microscope, let us say on a surface how the growth looks like, you can see there are colonies and there are less growth areas. So it is not just when we say film, it is not a uniform film. When you say thin film in a chemistry or physics, it is uniform film. But in a biofilm, when you say biology film, means it is like a microbial film. It is equated to a like a city. You have a pockets of dense growth. There are less dense growth. That's how something looks like. So there is a colonies, and there are areas where only single cells can. But this all involves that there is a sequential time-bound growth. Initially there is attachment, then attached cells grow into bigger colonies, and there is also migration of bacteria from one colony to another. There is a lot of chemistry is also going on, signaling how these cells become colonies, and then how to what extent they grow, and then there is some detachment of these colonies or dispersal. Okay. So, and this kind of growth I said in the nature, but that can be also formed in the lab under the conditions you design. Let us say I have given a surface, this red colored surface is a hydrophobic substrate, hexadecane. When I give this hexadecane and put it in the water, it forms like a bubbles, small particles, and over which you see how the microbes attach and grow on this hexadecane. Hexadecane is highly hydrophobic because there's a carbon chain length, there's highly hydrophobic. But even then, bacteria attach and grow and form a film over time, and this gets smaller because they start utilizing it. Okay, some cases they start utilizing it. Some cases this will be just a surface or substratum for the growth of the microorganisms. So there are two distinct differences I would like to mention here. One is in nature, bacteria are most of the unicellular, bacteria and some of the unicellular eukaryotes like microalgae, bacteria as well as cyanobacteria, which comes under bacteria category. There is also unicellular microalgae like even diatoms, okay, even chlorophyce, chlorella. So they can grow like, they prefer to grow on surfaces. So that is the predominant growth that has been established. And not just that, they grow on surfaces and they grow as a communities along with their own kin, also other different genus and species, etc. So that gives them better advantage to live in the nature. So it is not just nature, they grow like that. In our health, let us say dental, yellowing of the dental, teeth. Also the open wounds, and then even the implants that has been put into the body. So they become surfaces for the microbes to grow on. Okay. So there are various infections where the microbial growth or biofilm formation is prevalent. And I said about environment, I said about the health, and I am coming to the again industry. So we use water, right? There are pipelines, we take water every day to home. So there are water treatment plants. So on these pipelines, this again, because water has a life in it, microbes live there, and they colonize the surfaces, they grow as a biofilms. The issue in water treatment lines is that there are also pathogenic bacteria. These biofilms can become pockets of, places wherein this pathogen, like Legionella and all, can grow there. And then I also said the life sciences in BRC, why it is important earlier. That's because any power industry, they require water for cooling purpose. So large scale water, actually large amount of water is drawn into the cooling water system to take away some of the waste heat from the power plants. So because I said life is there in water, natural waters, be it a river water or it is sea water. So there are microbes. Also there are like larvae of higher organisms. Once this water goes to the cooling water system, they colonize the materials in the system and they make their own home and they start living there, which is not desirable because this can lead to reduction in the water flow. Also it can actually impede heat transfer across the condensers and heat exchangers, which is actually directly linked to power production. So power plant authorities doesn't want to reduce the, they actually, it is an operational challenge for the power plant operation. So what they do is, they add some amount of chlorine, they also add some amount of surfactants, other treatment to reduce the biological growth in the cooling water systems. So likewise, many industries, even let's say shipping industry or food industry, there are pipelines, we see this kind of a problem. So I mentioned planktonic growth in the beginning, right? In the lab, what we see. But I also mentioned biofilm. Biofilm means there is a growth on a flat surface, or surface could be like a three-dimensional in nature. Okay. But there is also growth. We call it as an aggregate. That means microbes have the ability to attach to each other and then form a clumps. So that has been observed in water, also in lake water as well as the sea. So there are different names are given, marine snow and then lotic aggregates. Even in the health bloodstream, this has been observed recently. And so aggregates have been observed. Biofilms are known for quite some time. But the recently also there is the observation that even in the health or human body, these bacteria live like aggregates. So why we are talking about this bacterial biofilms or aggregates in health? Because when a bacteria lives in a biofilm or aggregate, which becomes much more tolerant, 10 to 100 times tolerant to the antibiotic. So it becomes a problem for the clinicians or the doctors to devise an antimicrobial strategy. So then I'll come to another part. There is also like one is the tolerance or resistance, and then there is an antimicrobial resistance. Tolerance and resistance contributed by their growth, like biofilm. But there is also like there are organisms which develop resistance to the antibiotics. So which is known as antibiotic resistance or antimicrobial resistance, AMR, which is prevalent and talked about these days, because that is not a good thing. Because any drug you develop and these microbes develop resistance to it, and then there is again a problem in utilizing these drugs for the treating them. So in that context, we work on one side to develop or make formulations which can actually prevent the growth of microorganisms or biofilms. At the same time, they also do not allow evolution of antimicrobial resistance. So to mention one of the products we have developed, that is it is actually for wound healing, which is based on a nitric oxide releasing fabric or band-aid, that actually what it does is nitric oxide is used in our own bodies as a signaling molecule. Here nitric oxide also has an antibacterial activity, so which can reduce the growth of microorganisms. So that concept has been utilized to make this band-aid. When you apply to open wounds, so it releases the nitric oxide locally in a controlled release manner, so that it can prevent the growth of biofilms. As a result, wound healing becomes better or faster. So that has been mainly utilized or used, useful. Market is ready for release, but we also have patent also, product is ready. One of the companies is releasing into the market, and after all the clinical trials. So, and what we envisage is that is useful for treating mainly in the case of diabetic patients, because there the healing becomes an issue, foot ulcers and so on. So I will come to the other side of these microbial communities, how they are useful to us in let us say making our environment cleaner and water better for the future generations. So there is like the sustainable development is nothing but the development, but at the same time we preserve the resources, be it water, soil, or any other resource for the future generations. So that is there are also United Nations has brought out 17 goals, which are called global goals or SDGs, to make sure that the sustainable development is achieved. So these are different sustainable developments. We don't need to go through them, but what I would like to mention is water is a critical component in many of these sustainable development goals, because in the sea, let's say life under the sea, if there is a pollution, the life gets affected. There's a lot of talk nowadays about plastic pollution, microplastics, nanoplastics, and so on and so forth. At the same time, emerging pollutants. Nanoplastic is one of the emerging pollutants. And then the used water, let's say water is used at homes. We use, let us say, approximately 500 liters at home. Each house requires about 450 to 500 liters. And after usage, where it is going into the drain. So if that is not treated well, it is going into natural water bodies, or it is going into the groundwater. Again, we depend on natural waters or groundwater for our piped supply. If the used water is not treated well, the same it is a cycle. So there is a requirement for us to treat the water which has been used by us. So it costs us. It costs us as well as the industry if the industry is using the water. But so that has to be properly managed by some kind of treatment. So there are different kinds of wastewaters. Mainly these are important because the houses we use large amount of water, and then industries also, because industrial wastewaters are more polluting than the domestic wastewater. And then the domestic wastewater, there are different nomenclature. We don't need to bother about the nomenclature. But let's say we use water for bathing and except toilet. Because toilet is the place where actually our gut microbes enter. That means our pathogens, not just gut, whatever excreta, which carries human pathogens, is the problem when we combine the normal water with the toilet. Otherwise, let's say other domestic uses, be it bathing or washing, which does not actually involve a lot of pathogens. So typically, if you see, this is the kind of used water we see at homes. So drain, and you must have seen open canals in Vijayawada too. So which carry a lot of this domestic wastewater and also a lot of foul smell. And then there will be breeding of the mosquito. And then of course, this is the main problem why we need to treat it is because the pathogens. Issue is the pathogens. One of the primary concern is human pathogens. The pathogens from here, it will go to the natural water. Finally, we will have a waterborne disease. So the pollutants are defined in terms of different categories like the organic matter, nutrients, and then pathogens like that. But there are also hidden resources. Nowadays, it is already realized that wastewater can be a very good source of water for meeting the demand supply in many of the cities. So, for example, Chennai, city like Chennai, they treat domestic wastewater and use the treated wastewater for the industrial use. Similarly, many places this treated wastewater is used for, of course, gardening it is used. Apart from that, even toilet flushing and other uses it is being used. In case of Singapore, they treat the wastewater and they reuse for drinking. So they apply our treatment, finally it is going for the drinking purpose. So there are various problems if we don't apply proper treatment. So if you take Indian scenario, we have only treatment capacity for treating only 30% of the wastewater that is generated from homes. I'm talking about the bigger cities, capitals, like not at Amaravati. Let's say I'm talking about class 1, class 2 cities as far as the government of India is concerned. That is like Chennai, Hyderabad and all that. Because even rural we are not covering all these things, because wherever urbanization is there, this problem is become more complex, and only 30% we have a capacity for treating. And rest where it is going to the either to the sea or to other discharges like land or water bodies. So why we are not able to address even the developed countries like Europe and US, they have a coverage of up to 90%. But developed nations, we have an issue. That's because if you want to treat any water, we need a plant. We need an infrastructure development. So, and we also need a operating cost. So there are two components that decides. That is where even companies doesn't want to invest so much money. There are technologies, but there is a cost. If you want to build a treatment plant, STP or ETP, there is a capital cost for the plant. There is also an operating cost per liter of water that is being treated. So these are the main reasons. That is why this difference exists. So you see why I am talking, because this treatment doesn't come under biology, right? But still I am talking. I will come to that point why biology plays a role, or how biology plays a role in the treatment of the wastewater. So in any treatment plant, sewage treatment plant, so you see different components. So you see a bigger picture I have made for biological treatment, because this is the one which removes majority of the pollutants. That is the one does most of the cleaning job, because most of the pollution I mentioned, the organic matter or nitrogen, phosphorus, all that they are nutrients for, they are food for the microbes. Okay, and it is also that it is happening in the nature. So we are only taking that and accelerating the process. So there are different technologies available, I mentioned. So this is the age-old process, almost like a century-old process. But there are new processes, because I mentioned cost and for plant building, treatment plant, also cost for the treatments. So why we need technologies, new technologies to reduce those. So we need to improve treatment. At the same time, the technology comes with a lower capital cost as well as the treatment cost, so that can become more affordable, so that we can improve the infrastructure. Affordability increases. So because of that, the developments have happened. So this is the latest one. I will be talking about how the bacterial granules, what are those, how they help in improving treatment, also reducing the costs. So I mentioned activated sludge is like a 100 years old process. So which was developed in 1914 in London. But there are many of these plants all over India and many places across the globe. But it requires a large chunk of place. Also, it requires infrastructure and then cost for the treatment. So what it does is, this basically there are two tanks. One tank for mixing. I said earlier, aerobic and anaerobic growth, right? So that is what is done here. The mixing is done to provide oxygen, so that microbes grow fast and they oxidize the organic matter using oxygen. So microbes grow faster, so they double their numbers. Typically, E. coli requires 20 minutes for doubling their numbers. Okay, so something similar. So we need one tank for treatment, another tank for the settling these microbes, so that the clear water will be there. That water will be released into the either canal or to the treated water, or this water can be reused for other purposes. Okay, so but problem is this configuration, it doesn't remove ammonium and phosphorus. So ammonium and phosphorus are agriculture. If you have anybody with agriculture background, we use fertilizers for the plants, right? So they are not DAP or urea, etc. So there we are supplying ammonium and phosphorus there, fertilizers, because to accelerate the plant growth. When we don't remove ammonium and phosphorus from the domestic sewage we use and generate at home, it will go to the natural water body and it will cause the algae to bloom there. So algae, they take up these nutrients and they go faster. So as a result, we see greening of these water bodies. So we call it as eutrophication. So because of that, the fish will die. Fish will not get oxygen. So which is not a good thing. So what is needs to be done? The treatment should remove ammonium and phosphorus also, apart from organic matter. Another thing is why we need two tanks. We can separate it out here itself, right? After treatment, we can allow them to stand and separate. But problem is this microbes, no, they grow like this. If you look under microscope, you see like lot of filaments, lot of filamentous structures. Okay, there are also cocci or rod-shaped bacteria, but there are also large filaments. That means if there are filaments, they cannot make a strong compact particles. So as a result, once these are like fluffy, we need more time for them to settle down. So that is the very reason we need a separate settling tank wherein we allow these flocks to settle down to the bottom. So I said about this already, activated sludge. Okay. So then there is something like attached growth, which I referred to biofilm. Initially I mentioned biofilm. That means you provide this kind of surfaces in the wastewater treatment plant. Microbes attach and form this kind of growth. Okay, attached growth. And they do the cleaning job. Here, all these cases we are referring to all sorts of microbes, not just one bacteria, two bacteria, or fungi. Okay, because the wastewater is complex. There are multitude of pollutants there. There is like organic pollutants, which is measured as chemical oxygen demand or biochemical oxygen demand. There is also ammonium, nitrate, nitrite. There is also phosphorus. Okay, pathogens. So we need a multitude of microbes. So if you use one microbe, no, it may remove organics, it may not remove ammonium. So that's why we need a community comprising of heterotrophic bacteria which oxidize organic matter. We also need a nitrifying bacteria which convert ammonium to N2. You know, our air has a N2, right? Nitrogen, oxygen, nitrogen. That's not a problem, but ammonium is a problem. So ammonium is converted to N2 by microbes, and then N2 goes to the air. Okay. Similarly, phosphate is accumulated by the bacteria. So there is another development after biofilms. So wherein these microbes can be made into bullets or beads. So it's interesting actually. See, let's say a microbe is of 1 micron in size, which we need a microscope to view them, right? But if you provide conditions and they grow in few days as bigger particles, we can view with the naked eye. It can be like few millimeter particles, 1 millimeter, 2 millimeter, 5 millimeter sized particles. Because it's a natural growth accelerated in the bioreactor. Okay. So what is the advantage? Once they become size is slightly bigger and they are compact, they separate out much faster from the treated one. Also, these microbes, when they are in the granules, they perform better because they are hidden in the matrix. They don't get intoxicated by the water pollutants. If there is a high pollution, they can still work within the granules. So, what are these granules? I said bacterial granules. Okay, they are made up of mostly bacteria. And apart from bacteria, they produce a polymer, which can be exopolysaccharide, let us say alginate or xanthan. Also, they produce some proteins, adhesins, to make contact with other cells. So as a result, no, they make a aggregate that grows into a bigger particle, like a granule. So this I mentioned, activated sludge, no, separation takes long time. So we need a separate tank for it. While granules, they settle out much faster. Okay. And if you look within one particular granule under a microscope, we can see this kind of structure. So there are like tiny aggregates within the granule. So you see each is one cell. If you see here, no, there are cells tightly bound with other cells, other bacterial cells. So this is a scanning electron microscope showing how these bacteria are attached to each other within a granule surface. So there is for cultivating these granules, we need some kind of a container. That's what we call it, bioreactor, right? So sometimes fermenters are used in the lab. So bioreactor, in this case, we call it a sequencing batch reactor, SBR. Basically, it is nothing but to simplify it further, it can be just like a one tank. Okay. So what I do is I fill it with the water to be treated, and then give some time for mixing. And then reactions are done. I will stop the mixing. Means I will stop aeration. Then it is ready. There is like whatever dense particles will come down. Then the top treated water is decanted. Then this is ready for again another batch of treatment. So that's how treatment is done. And I also mentioned is the growth process, because it's a living system we are talking about. There is a time scale involved. So it comes initially we get small aggregates that will eventually grow into bigger particles. Okay. So what it does, so it converts organic matter. Let's say we use leftover food at home. Okay. Also some undigested food from the humans, fecus. All that will be converted into carbon dioxide, which will go into the atmosphere. And then these cells, while doing so, they double their numbers, microbes. So that's why we generate fresh biomass. So then ammonia gets converted to all the way to nitrogen. So there's a nitrification, denitrification processes that will convert ammonium into nitrogen, which will also go into the air. And there is a phosphate also in the domestic wastewater. The phosphate case we cannot do much, but these microbes are some microbes. We call them as phosphate accumulating organism or PAOs, polyphosphate accumulating organisms. What they does is there's a process by which they accumulate inside and make polyphosphate. So basically different processes are involved like biodegradation, biotransformation and accumulation, things like that. And there are also some examples I just wanted to show you because there is like a textile industry in many places, no? Effluent treatment is an issue. But there also they will have a treatment plant. There is also biological treatment part of it where microbes does the conversion of textile dye into non-colored product. We call it decolorization. Here you see the color of the dye. The color of the dye, and then there is no color after some time. So this is all done by the bacteria. It is not just the dye. There are many toxic metals. Microbes can interact and then convert into non-toxic forms or non-soluble forms. That's how the heavy metal pollution can be handled by the microbes. Some cases like this, chromium or selenium, tellurium, or many of them are toxic to the microbes. But there are microbes, they convert them into non-toxic forms. These are like either non-soluble or non-toxic to the bacteria. This is some example, just wanted to show. These are normal granules. When I give a selenite or a telluride, they convert them into another non-soluble form, which is colored. Okay, this coloration is an indication that the selenite is converted into Fe0. Okay, so water is free of these metals by then. So this is again bacteria when they on a plate, agar plate. When they convert? No, the color is because of the metal. And then if you look further into that, the colored product, we see formation of these small white particles, nanoscale selenium 0 particles on the surface of the granule. So I will just give you some example of one of our work that has gone into the field, just to give an impression that the lab work, how it goes into the field, how it gets translated into the field. So we have been working on these granules for quite some time, from let us say 2003 or 2004. As part of my PhD work, I started cultivation of these bacterial granules and their technological applications in wastewater treatment or bioremediation. So as part of my PhD, I did some work. We know that these granules can be formed in the lab, and then they can be easily cultivated in the lab. And we know some amount of mechanisms how they are formed. And also we did some work to show that they can remove some pollutants in the defined conditions. The work was taken up because the granules can perform better than the existing methods. That's one reason we took up this work. Also, another reason is that nuclear establishments, they generate some effluents which will contain high amount of pollution. So high concentration of nitrate or high concentration of tributyl phosphate, okay, some amount of sometimes urine, okay. There are few reasons why I choose this to work on, or why I choose our group. One is, first thing is, this work is happening elsewhere in the Europe. Second reason is, this work nobody is doing in India. Third reason is, we wanted a most rugged system that will work for more tougher pollutants. And fourth reason is making millimeter sized particles from a micro-sized bacteria was interesting. Process of making them, the biology of making them was interesting. So this is how the process actually development looks like. It's a simplified development process. So this is what has been already done by others. And then we choose to make this kind of particles from the microbes present in the water itself. Let us say it is seawater or the wastewater. And this approach we have taken. And then we made this kind of beads in the lab. So there are like when we made these beads, it was exciting. So then we thought it has a lot of potential when it goes to the society. When we were working for our own effluents, because nuclear, we cannot patent anything related to nuclear applications. That's not allowed. But at this stage, we found that this can go to the society for normal wastewater. Then we went into like IPR, and then we made a patent, Indian patent. And because our interest was to commercialize. Okay, that's the reason for patent. Otherwise, there is no need for the patenting this process, especially. Because we wanted to commercialize, then we went for the patenting of the process. Then after patenting, then we started, let's say lab anyway we were doing, but not with the real wastewater. So because we wanted to take it to the field, so we have to work with the real wastewater, that is sewage or domestic wastewater. That cannot be handled in the lab. Most of the labs we don't handle those. So we have to go to the field where the STP is there or field sewage treatment plant is available. So where we have actually there we have set up small scale reactors like this, which are in-house made with very cheap materials, and then there is a pilot scale reactor. So 20 meter cube means 20,000 liters. This way, right? I said this is about 150 meter cube means 150,000 liters, and this plant is like a first full scale plant we have built, and this can cater to roughly 320 houses. So when I say one house, we have said that one house generates about 500 liters. If you calculate, it will come to roughly 300 to 320 houses. These are again pilot scale plants, different capacities. So in pilot scale plants, what we demonstrated growth of these particles with the real wastewater under field conditions compared to the activated sludge, which is much more dense and able to remove pollutants effectively. We also demonstrated treatment by giving just 4 hours of time. Within 4 hours, the treatment is complete. We have a dark this color water. Within 4 hours, we get a clean water. Let us say. Because this is required, because parameters I mentioned, because these are the parameters our pollution control board requires. So that's why I am mentioning only those parameters that are mandated by the pollution control boards. So I mentioned 150 meter cube plant earlier. What does it actually contain? So this is the room where equipment are kept like control panel, and there are some lower equipments, things like that. And there is also a bioreactor. Here I was referring bioreactor, green color tank is the one. That is the place where this granulation takes place, also treatment takes place. And then after treatment, there is also some kind of filtration that is done for disinfection. So this is the simplest process of how it is done. In 4 hours, the whole process gets completed. And then we see, this is the kind of field level demonstration. You see, I am showing earlier the big granules, big nice pictures. But when it comes to the field, the picture becomes very small particles, right? So there are still challenges translating what we see in the lab to what we achieve in the field. Okay, but the interesting thing is we were able to treat it well. So we are able to, the function is it's good. Although we are not able to make big granules as we see in the lab, because in the lab we provide ideal conditions for the microbes to flourish. and then good food, defined food, etc. When it goes to the field, there are a lot of variables. We do not have a control on the field, what comes into the plant, treatment plant. Similarly, there is energy also matters here. So we have to have a balance. Okay. So this is like a hypothesized the picture of a granule, a single granule. If I take, how do I see these microbes inside the granule? So I said earlier that I see different types of microbes within the granule. So I give oxygen outside, right? In the bulk liquid only we give oxygen. So we expect the green region aerobic bacteria to grow. So that include nitrifying bacteria, also heterotrophic bacteria, which oxidize ammonium as well as the this also, organic matter. But inside we will see some kind of anoxic or anaerobic conditions where we see the denitrifying bacteria or the anaerobic bacteria, some polyphosphate accumulating bacteria, things like that. And what the matrix is made up of? Because the cells produce polymer, which is a complex matrix combining of various polymers, including in carbohydrates, proteins, and some amount of lipid. So some amount of alginate has been observed in the matrix. So this is how the BOD organic matter gets removed by bacteria. They oxidize and convert it into carbon dioxide. And there is also ammonium. I mentioned that there are two groups of bacteria which does nitrification and denitrification. Within nitrification again there are two groups: ammonium oxidizing bacteria and nitrite oxidizing bacteria. Okay. But what essentially we found is that our process is much more efficient than the existing processes in terms of removing the ammonia from the wastewater. Phosphate requires a special attention. Among all nutrients, phosphate is limiting in natural waters. So when there is not growth of algae or whatever, because there is a limited amount of phosphate is available in natural waters. But sewage have 4 to 5 ppm of phosphorus, and most of this treatment plants does not remove phosphate. As a result, there is a plant, there is infrastructure, plant is there, we are spending money, but because of ineffective treatment, we still see the problems. Another interesting thing is phosphate is required for agriculture, and most of the phosphate we import from China and other countries because it is geographically unevenly distributed. Phosphorus. So phosphorus is also one of the critical elements listed by Government of India and other countries. We have about 20 and odd critical elements which are required for sustainable growth, also making these green technologies. So recent talk, people are talking about rare earths. There was also talk. Rare earths, strategic elements like uranium, also like something like phosphorus. But but wastewater has a phosphate. If you are able to recover and then it goes into the field for agriculture, we can at least reduce that burden, import burden of phosphorus. So that's what actually is done here. So these microbes actually, one group of bacteria, they have ability to, they uptake phosphate and make polyphosphate. So that will go to the field and there actually polyphosphate becomes phosphorus, and that can be taken up by the plants. The phosphate, there are methods available for removing phosphate by chemical methods. The problem with the chemical methods, we have to add chemicals like aluminum or iron salts. And though we remove phosphate, that is not a sustainable approach. Because it is the chemistry, everything is defined. But the thing is, the whatever approach is not sustainable. Also, it is a costly process. But the biological process, sometimes there are variables. By biological process, you will not, you see some variation in the process. But if you are able to establish and remove phosphate, because you are making it in a sustainable way, that phosphate will go to the field. And pathogens are removed in the treatment. There are various ways by pathogens are killed. First thing is aeration. When you give oxygen, most of these pathogens are tolerant, they are unable to bear the oxygen concentration. They will get killed. Okay. Also, there is a disinfection after biological treatment that is being applied. So, now I almost come to the end. Yeah. So, then there are, I said, alginate and this group. There is also concept like waste to wealth, waste to wealth. That could be solid waste or liquid waste or even the e-waste. Okay. In the case of here, I am talking about the liquids, liquid wastewater. So the nutrients are important. Apart from nutrients, when these granules, when they treat, they are making a lot of polymer, and that polymer is getting accumulated in the granules. So we found that that contributes to almost like 40%, and this alginate has some commercial applications. So it is like this kind of approach can recover some of the cost we invest on treatment. And there is another thing. So far I mentioned about only bacterial granules, but there is another thing wherein we can combine bacteria with algae. Algae is called a sustainable approach because algae respiration, they generate oxygen that can be used by the bacteria. As a result, total we can reduce the aeration in the system. And these are some of the plants that were inaugurated by our top management, Secretary Atomic Energy. These are the pictures of some of the plants. So what we did is, after making all this pilot test and then full-scale demonstration plant, we actually, because as an ERC academic or research institute, we cannot make these plants. So we can only make one plant as a demonstration project. Then we have actually floated it to the companies, asking them to come forward, and as a technology, there are almost like 37 companies so far, including large companies as well as MSMEs, who have signed agreement, and they started implementing this technology, and they built almost like 15 to 20 plants at different places during the last 4 years. So these are some of the plants again in Shirdi. One case example I would like to mention. This is one of the event that has happened in Prayagraj, and it witnessed large enthusiasm, and then the place was really crowded with the pilgrims and other participants. And what they done is to maintain sanitation there. So they have a tents, large number of tents for the sadhus and others. They were going and staying there. And in the tents, they provided these kind of toilets. Okay. And then for treating this, because this toilets have to be emptied and then treated and then discharged. So for that, they floated a contract given to one of the vendors, and they choose actually our technology, one of the technology. Ours is a only technology, and then vendor has supplied 3 plants. So each with a 500 meter cube capacity, 500,000 liters capacity, and these plants were operating there for almost 45 days during the Kumbh period, and treating the sewage, and then releasing that sewage into a canal that will finally go into the downstream of the Ganga. So this is how the treatment plant, and these are the tanks. They collect the sewage from the temporary toilets and then discharge to the treatment plant. It is treated biologically, and then there is a ozonation at the end of biological treatment, and then finally that is disposed of. There is some of the news items related to that. Yeah. So this is one thing. I just mentioned in the initial talk how I entered BRC. BRC has a OCES exam. So most of the entry into Baba Atomic Research Center goes through this kind of recruitment. So it is very transparent kind of recruitment process. So that involves written test. I think written test is conducted even in Vijayawada for the postgraduates in life sciences or physics or chemistry, or graduates in engineering. And after that, interviews are held in Mumbai. And then we also have a Homi Bema National Institute, which comes again under Baba Atomic Research Center. So there is a PhD program actually. So they annually take the students, they conduct their own exam, and then the fellowship is given by the constituent institute. So it's not that a student has to come with their own fellowship. Yeah, thank you. Thank you very much.
Before concluding, I request the audience to interact with our resource person. If you have any doubts, you can come forward. Any doubts from the participant side?
Anything you can ask.
You can ask also how to prepare for the OES exam also.
OCS exam is for the masters, but once you are completing your masters, you are eligible. Yes.
Okay, nobody is asking. I'll ask a question.
I won't ask difficult question, man.
No, it will be difficult because I did not understand. I don't understand. I'm asking difficult. So, the granules that you form, how did you initiate the granule formation? What was the mechanism? What is the purpose of granulation?
Yeah, there are two things. One is purpose I tell, other thing is difficult to tell you. So purpose is presently activated sludge is used. That requires two-time configuration for treatment. One is for treating, another is separation. Basically, biological treatment involves treatment and separation. So here both are combined into one tank. Okay, so that requires faster separation. Once they become bigger particles, they settle fast. That's one. Another thing is once they are compact, we can maintain more MLSS, typical term that is used, or biomass levels in the reactors. So instead of 2 or 4 grams per liter, 2 will go up to 8 to 10 grams per liter. So that will improve efficiency as well as the fasterness of the treatment. Third is they become more inside cells. No? They don't really expose to the toxic levels in the bulk liquid. So they can tolerate and do the job. Coming to mechanism is we know a little bit, but still a complex process. It's a gradual process. Yeah. So I would say we don't know much. Yeah.
I have one question, Dr. Nachman.
Yeah.
I would like to know much about, you mentioned that there is a kind of an aggregation formation inside the human body. Is there any specific locations that you observe in the human body? And normally we have normal microflora. We should have them for our good digestion or vitamin B12 production inside. If it happened because of the pathogenic bacteria, it is very difficult to treat that.
Yeah.
I mean, I would assume that in the excretory system, do they form any kind of biofilm, or is there any preferentially they form in some organs?
Actually, this biofilm formation is an issue. Let's say both topical as well as the systemic infections. Okay, even the Pseudomonas aeruginosa, which is responsible for the pneumonia and lungs issue, also uropathogenic this one, and dialysis side also, and implants. So implant colonization and replacement. Anyway, wounds, it is well known that will cause the problem, the open wounds. So, but aggregates thing is a recent one. they have observed in the systemic infections formation of these aggregates. So the challenge is the treatment, they face a problem. Conventional treatment doesn't really help to mitigate once they are in biofilm form or aggregates. But aggregates, as I said, it is a little information as of now. There are only few reports.
I guess these implanting materials nowadays, the biocompatible materials are developed that does not facilitate the formation of the things. So, right? For example, stunts and all.
Yeah.
If they facilitate the formation of bio,
Yes.
that would be really dangerous.
Yes,
I think they always have advanced materials
Yeah.
that would not facilitate the formation.
exactly. Yes, you're right. Yes. That's also still actively pursued, both for health, antimicrobial, even for environmental applications also. This antimicrobial surfaces are this.
Good morning, sir. Hello.
Yeah.
My niece completed MTech embedded systems with BITS campus, Hyderabad. Even though she has placed in Western Digital for 35 lakhs, when I asked what is your first priority, she said that I want to clear the examination in bar.
Okay.
So most of the students from IITs, NITs, and the other agencies, they are looking for bar. May I know what is the reason for that, sir?
Engineering side, right?
Yes, exactly.
Yeah. What is the reason why they are looking for VRs? Anyway, glad to note that they are interested in joining it. Yeah. The thing is, what I see is there is good and bad side. Good side is when they join as SOC, we call it as 1 year training. After that, they are joined as scientific officers. The good thing is that they have enough freedom to work. So they have freedom to work. There is enough funding to support their ideas. At the same time, they are also allowed to take up further career development. Okay, that is one thing. So it's not that, although it varies from lab to lab, but generally they are given good support by their bosses. At least initially. Later on problems may arise, but initially, and they can do what they want. So it is the bossism in terms of doing science is lesser, definitely. A bad side is that sometimes too much freedom is not a good thing, right?
Yes, sir.
Yeah. So that thing. Yeah. So otherwise, other government related issues are there. There is also good thing about pay compared to other government agencies like DRDO and including BRC, DAU. The space they have an additional pay component depending to their performance.
But most of the students have a perception that the work is related to physics and chemistry. But
Physics is very, very important. But physics, yes.
most of the engineering students are trying to let into the box. That's why I wanted to know the reason.
No, but actually, from community-wise, if you see, engineering is more in years as such.
Thank you, sir.
Yeah.
So if there are no more questions, we'll move forward. Thank you very much, sir, for your interesting and informative lecture. And it was also very simple because as a chemistry person, I could also understand it very clearly. So I'm going to sum up in a very few words. Sir has started the lecture with the growth of microbes in different environments. What are microbes and how do they grow in the human body and also as well as outside. And also we could understand the two sustainable goals, 6 and 14, clean water and sanitation, life below water. So, and we also learned about domestic wastewater characteristics, the various pollutants and how to treat the domestic wastewater and how they are being treated in various cities like Chennai also. Then different various treatment technologies of wastewater and various plants, the nitrogen and phosphorus removals, textile dye decolorization, and microbial assembly, how bacteria look in a granule, and also the various HB, SBRs, STPs plants, the pathogen removal, and the future directions also, waste to wealth, and especially we heard about liquid wealth. And sir also gave an example how HGSBR plants were set up in Mahakumbh 2025 at Prayagraj. Thank you very much, sir, for giving us a wonderful lecture in this area, role of microbial communities in sustainable development. I request all of you to give a big hand. Now it's time for felicitation. Sir, I request you to please come forward and occupy the chair. I request Director, sir, and HOD Srinivas, sir, and the faculty members.
వాళ్ళకి ఏపీఎల్ సెట్ చేసాము ఆన్ చేసి వస్తాం.
సార్ లేదు లేదు అక్కడ ఉన్నాయి వాటర్ ఉన్నాయి సార్. నాకు వస్తే అక్కడ ఎలాగో అక్కడికే వెళ్తాను సార్. మైక్రోఫోన్
Welcome back once again to the two-day conference, Trailblazing Pathways in Life Sciences. We have done our first session by our chief guest, and now we call upon for second session. Chairperson for this session is Dr. T. Ravikiran Garu. Please occupy the stage, sir. Next, rapporteur for this second session, G. Samrat Krishna Garu. The stage, sir. Now I take privilege of inviting today's talk, guest of Dr. Nagmohan Garu from BITS Pilani, Hyderabad campus. Sir, title is Mewers, a new class of variants with a potential for disease and susceptibility. Now I hand over to the chairperson for session 2 by Dr. T. Ravikiran Garu.
Good morning, everyone. I am Dr. T.S. Ravikiran from Department of Computer Science. It is a great honor to introduce today's speaker, Dr. Nagamohan Pomo, Associate Professor at BITS Pilani, Hyderabad campus. With over 13 years of experience in both teaching and research, Dr. Pomo has made significant contributions to the fields of biology and genetics. He is currently teacher a wide range of subjects, including general biology, genetics, stem cells, and regenerative medicines to students across undergraduate, master's, and ME level. His teaching inspires the next generation of biologists and researchers. In addition to his teaching expertise, Dr. Pomo research focuses on critical areas such as rapid detection of drug resistance, PB, the role of gen expression or supported gen expression in neurodevelopment disorders and the development of advanced genetic models. His projects are supported by the leading institutions like the Department of Science and Technology, DST, and the Department of Biotechnology, DBT. Dr. Pomo's work bridges scientific inquiry and practical solutions to pressing health challenges, and we are excited to have him to share his insights with us today. Please join me in welcoming Dr. Thank you. Thank you all for giving me this opportunity.
Thank you. At the outset, I want to congratulate BB Siddhartha College for 50 years anniversary, and hopefully, even if I'm not living through, wish them for the next century, you can say, of their endeavors in teaching good education and imparting education to students. And honestly, I felt very happy hearing about subjects like zoology and botany. And in my own, it's a very personal view. I never regarded, if some PhD candidate comes with a MSc biotechnology, I take it with a pinch of salt. My eyes light up when it is botany or life science and zoology. So again, I would like to thank all the organizers for giving me this opportunity to visit this campus, see it, and thank you, madam, for all the coordination, the director, the principal, the vice principal, Srinivas Garu, the other Srinivas Garu, who is a non-teaching staff who has taken care of us. It also gave me a chance to meet my friend after almost like 2.5 decades, Shiva here. So before I start my topic, please don't get overwhelmed. I understand that all of you are undergraduates. I'll try to keep the topic simple. Don't try to look into and find out what are the details of the slides. And one more thing I want to say is that don't even be bothered about whether you have understood it completely or not, because I don't think I have understood it completely, and that's why I'm presenting this. So, before I get to the presentation today, these are my students who have contributed immensely to our lab productivity. And this is our group which has graduated, and you can see me also there. So with many thanks to them, I want to introduce a very simple logic that is related to life. So if you take different cells of our body, all of them are the same in terms of genetic material. But yet, each cell has its own unique property and unique function to carry out, right? So the problem that I'm trying to present here is manifold. As far as pure biological sciences is concerned, particularly in the context of genetics, how is it that each cell carry a different function despite being genetically the same from another type of cell? So they all have the same DNA sequence, but for example, a skin cell would produce melanin, and therefore it's a skin cell. Red blood cells produce globin, and the neurons have specific activity, and the same way the muscles also have specific activity. So it's estimated that about 25 to 30,000 genes we have. The number may be even less. But the take-home message here is, in any given cell, many of these genes are not turned on, which we call it as expression. About only 1/3 of the genes are expressed in a body cell. And that unique set of 1/3 defines the property of the cell. And even if we see what is common, what is not common, you all have read about processes like glycolysis, Krebs cycle, and all those things. Even within this 1/3, which is something like 10,000 genes, a majority of them are of this category. Again, you don't see any differences between these in any living cell, right? So it's a very small number of genes that are going to make the difference, and therefore that particular cell is different from the other type of cells. So what contributes to that difference is central to our story here, right? So this is explained by a term called as epigenetics, where the DNA is identical, but the cells behave differently. And cells behave differently because different sets of genes are expressed. And the different genes are expressed differently simply because their epigenetic makeup is different. So I won't go into, there are many modifications of the DNA, the chromatin, and all those things. I'll only focus on the oldest, which has been discovered, which is related to cytosine. So addition of a methyl group at the 5th position in the cytosine makes it 5-methylcytosine, and like any chemical reaction, it is reversible. It can be removed and it can be added. But in a DNA molecule, when a methyl group is added, in the next round of replication, when it forms the daughter cells, the daughter cell acquires it. So there is a pattern of inheritance. So that is why, let us say, a skin cell will continue to produce skin cells and not any other cell type. But at the same time, it can also be reversed. So a skin cell can be converted to another cell. That's what we are seeing in case of in vitro reprogramming, where you all might have heard about induced pluripotent stem cells, right? So you can reprogram, but inherently epigenetic processes are heritable, at the same time reversible. And all of us have begun our life as zygotes, right? That is another example. We form a ball of cells around day 3, which is called as blastocyst, from which all different organs can be derived. And these cells, which are pluripotent in nature, at some point decide a group of cells will form a particular type of tissue, right? And that is epigenetically determined. Even there are reports, without me going into detail because it's not my main course of talk today, you can have cells responding to environment. For example, continuous smoking will alter the epigenetic makeup of the cells. So it reacts, it changes the epigenetic makeup, and at the same time, it is heritable. So I'm going to focus on DNA methylation, which is mainly pertaining to 5-methylcytosine, and even more specifically, if you look at this, we know that DNA is complementary, right? After replication, the daughter strand is produced by using the sequence information that is there in the parental strand. So what about the DNA methylation? So if you look at the cytosine, it can theoretically occur at 3 other nucleotides, including cytosine itself. So it can be CPA, CPT, CPC, and CPG, right? But if you look at the complementary strand, I'll just take one example. It is CA dinucleotide. The complementary strand is a TG, it doesn't have a cytosine. But if you take a CG, CG is symmetric nucleotide, dinucleotide. On the complementary strand, it will again read as CG. So the heritability is there. If that particular cytosine is methylated in the parental strand, the daughter strand, which will also have a CG dinucleotide at the same position, will get methylated. And if it is not methylated in the parental strand, it's not going to be methylated. So in this way, DNA methylation can be inherited, and therefore, by a larger definition, every other epigenetic pattern can also be inherited by the daughter cells. So not just the genetic information of the parent cells, but also the epigenetic makeup of the parent cell is transmitted to the daughter cells, with a few exceptions. The exceptions are, I don't know what the next slide is, the exceptions are mainly related to the early development and gametogenesis. So all of us inherit maternal and paternal alleles, right? There are a set of genes, about 150 estimated, which is a very special category of genes. These are called as imprinted genes. Now, we inherit imprinted genes just like any other non-imprinted gene, but for the next generation, their behavior is different. So I am inheriting my father's imprinted gene, allele, and my mother's imprinted allele. And for the next generation, even if my mother is a female, it is going to behave like it is a male, because I am a male. So suppose I have a sibling who is a daughter, sister, and I don't have a sister, unfortunately, but if she inherits, the same alleles for the next generation will behave specific to her sex. Right? So, for my son, whatever alleles I am passing through, for these 150 estimated genes that are imprinted, will behave as if they are coming from the male. And for my nephew, it is going to be different, as if it is looking like a female. The other examples of epigenetic phenomena is X inactivation. We know that females have 2 X chromosomes, males have 1 X chromosome. But if you look at the genes, the level of transcript that is made between a male and a female is equal. So that non-equivalence of X chromosomes is taken care of by inactivating one of the 2 X chromosomes in a female, so that the transcript levels come down roughly equal to the male. That's another example. So one wave of epigenetic reprogramming means that you are erasing all the epigenetic marks, including DNA methylation, and then a new set of epigenetic marks are made when the gametes are formed, depending on whether it's an oocyte or a sperm. That is one phase of reprogramming, where you have these in primordial germ cells, which will either produce based on the chromosomal sex of the individual, whether it is a sperm or a oocyte. After fertilization, there is another way, because you have got a zygote. Because of specific and special circumstances, the sperm is packaged in a certain way. Oocyte is much bigger than the sperm, right? And after fertilization, again there is a epigenetic reprogramming, right? So that purpose is to make the embryo capable of producing all the different types of organs that we have, including the placenta. So that second wave of reprogramming happens during the first 3 days of our life. We haven't yet implanted yet. We're just moving down the oviduct towards the uterus, day 3 and maybe 3.5 in humans. Before implantation, all of our epigenetic marks are being erased only to freshly start establishing new set of epigenetic marks. So we experience 2 waves of reprogramming, right? Naturally, one during gametogenesis for the next generation, and second, even before we are born. Fine. I'll skip this because I covered this already. This essentially summarizes that even if we are inheriting paternal and maternal alleles, they are going to behave differently. Right? This is called as the parent tug of war. And this applies to only those 150 to 200 genes. That number seems small, but that has profound effects. Just to give another interesting example, there is one gene which is called SRY in humans. It's a Y-linked gene. It's present on the Y chromosome. measures only 1 kilobase, it doesn't have introns. And interestingly, it is turned on day 10 only for 1 day. And for life, that embryo is made. Right? So don't underestimate the numbers. Sometimes they may have very drastic effects. So again, in the interest of time, I'll skip a few slides. So I talked about DNA methylation. Right? DNA methylation can be different in 2 different genes. One gene can be methylated, one gene can be not methylated. And this methylation can again happen in 2 different locations of the gene. All of you are aware of what is promoter, which controls the gene expression, and also within the gene, which we refer to as gene body. So in general, if you have promoters that are methylated, it's often associated with silencing the gene. And there is another area where methylation can happen, that is where it can be anywhere from the transcription start site to the end, which is called as gene body. And gene body methylation is associated with expression. So you have contrasting effects. Promoter methylation causes silencing, and gene body methylation often is associated with expression. We hypothesized a few years back that we were working on autism at that time, that among normal individuals, just by the level of methylation in each of the gene, autosomal gene, you can have different situations. So suppose these are all normal people. In normal people, of the 2 copies, one is methylated. That may be the normal state. For example, the X inactivation I told you already, right? But then you can methylate the promoter of this gene to increase the methylation and therefore turn off the gene. Again, taking some examples in cancers, many tumor suppressive genes have been reported where you have promoters methylated and turned off. The gene is there, mutation may not be there, but it's turned off. It's as good as not having it. The next example is that you have loss of methylation. So you have more than the required dose. That examples are also there. So we showed through these mechanisms that in certain autism cases, I want to take an example here, which will be applied to many other things that I'm going to discuss now. There are mainly 2 categories of genetic disorders. One is called as a single gene disorders or Mendelian disorders, where there is a single gene that gets mutated, and either in homozygous or heterozygous condition, it causes the disease. And most often, when there is a mutation, it invariably causes disease, like hemophilia, for example. If you have factor VIII mutation, it causes hemophilia. Or a beta globin gene deletion, it causes thalassemia, major or minor, right? Same way, if you see complex disorders, which we commonly encounter, they have a strong genetic component, but they also have non-genetic components. That is where the complexity is. And any common disorders that you take, whether it is susceptibility to diabetes, hypertension, mental health disorders, all these come in this category. And if you note, I have actually used the word called as susceptibility to diabetes. I'm not saying anything that it's going to cause diabetes, right? So these are caused by many genes, maybe a combination of environment and so on. So what we discovered is that in certain autism cases, we identified DNA methylation abnormalities in autism patients that turned down the level of expression of peroxidase enzyme catalyzing gene, which is catalase, and another gene which is novel, that's called as SCRH. Let's not go into the detail, but here the only way you can identify these genes is by treating each individual as unique. We wouldn't have found these genes if I have taken what is commonly different between normal people and commonly different between the patients. So here we took each patient one at a time and compared with all the normal people to identify what is that specific abnormality. And as part of this study, we also saw that about 1/3 of the genome showed significant amounts of differences in the level of methylation within normal people. That forms the foundation of today's presentation. In other words, in this room, whatever number of people are there, even if you are all normal with no mutations, epigenetically you are different from each other. And therefore, your susceptibility to disease will change. So, with that hypothesis, we wanted to now set out to find what are such genes that are carrying this kind of high levels of differences in the methylation levels between people, and what genes are involved, and in which ways the genetic studies indicate that these genes are associated with different kinds of conditions. So again, I won't go into the technical details. In a small pilot sample, which involved 45 cerebellum samples, so we identified nearly 2,000 genes that contain methylation differences within normal. There seems to be some correlation with the age, but I don't think it is significant. But about 50% are present in just 1 or 2 individuals out of this 45, which means that each individual is unique. And this is the frequency of distribution. So, on one hand, I am saying there's a vast majority where you have just 1 or 2 individuals showing that difference in methylation, which we use the term as methylation variant. But you also find some very low number which are present in substantially good fraction of the people, like about 25 to 30%. So these are some examples. Again, I want to just take a pause here. Today's life science research requires you to be an expert in mathematics, statistics, programming, and you need to know chemistry and physics also, because life defies the rules of chemistry and physics. That's why we are living. So to know what is right, you should know what is wrong. So these are just some examples. Don't go into great details. On the y-axis, I have plotted the methylation levels, and they range from 0 to 1. 1 corresponds to 100%. And if we just see one of these genes, the horizontal lines represent the averages. You have a bunch of people with low levels of methylation, and we also have some which are having high levels of methylation. So remember, there's a set of 45 people, and we have extended the study to more than 3,000 samples. And overall, we have identified 12,000 plus methylation variants present in more than 8,900 genes. That's a substantial fraction. But importantly, the trend that these variants are individual specific. It's not that it's going to be common among people and so on. So each individual is different from the other. And for getting more details into it, this involves 7 different tissues. And you know the primary germ layers, right? The ectoderm, endoderm, and mesoderm, to know how these are distributed. And what we found is that these variations differ from tissue to tissue, and it seems that the skin has highest number of variants. And this is one example. Incidentally, this gene mutations cause mental health disorders. And the blue bar here indicates the methylation variant region, and this is a promoter that contains a CpG-rich sequence. Right. And this particular gene has this kind of bimodal distribution. Bimodal means on one side you have a group which has unmethylated sequences for the same region, whereas the other one has highly methylated regions. They don't seem to be differently occurring at different portions of the gene. If we look at the methylation variants, they seem to be distributed not so significantly different across the promoters or the very first exon of the gene body and so on. But there's a clear difference between a group. I'm trying to avoid this term, but I will probably have to mention this. So 50% of the human and mouse genomes, and presumably other mammals, contain promoters that are CPG rich. And these are called as CPG islands. And the remaining 50% don't have such CPG rich sequences. And for those in chemistry, cytosine can undergo spontaneous deamination as part of exposure to DNA damaging agents and get converted to uracil, which is a foreign nucleotide for DNA and is fixed immediately by DNA repair. Whereas the 5-methylcytosine, when it is deaminated, it gets converted to thymine, which is a natural base for DNA. This is one of the mechanisms by which we accumulate mutations. There is a very efficient DNA repair system, but per each generation, we have, I think, 3 to 4 mutations per every 10⁻⁹ basis. So all of us carry new mutations to the next generation. That's part of evolution. So having this information, what are these genes? What are they associated with? So it turned out that these genes are either mostly significantly associated with the mental health disorders, different types of them, like schizophrenia, depression, bipolar, autism, and so on, but also many different types of cancers. We didn't focus on cancer for a reason. The main reason is that in the context of today's talk, we are more focused on discovering the pathways rather than dealing with the pathways. So it's again my personal opinion that at the moment we don't know what causes cancer for sure, but rather than we know what is happening in cancer. So we wanted to steer away from that, and of course mental health disorders is our research area of focus. So we looked at that and we were very happy that schizophrenia was among the top. And these are the different pathways with which they are associated with. Again, many of them, for whatever reason, we have lots of genome scale data. The mental health disorders and the cancer genomes share a lot of pathways. And there's also a debate on whether schizophrenia patients, some papers say that schizophrenia patients have a higher susceptibility to cancers, and some say the other way, that they're more resistant. But the fact is that there's a major overlap between the two. So again, these are just some network pathways, protein-protein interactions, substantiating our earlier findings. So most of the genome scale data today is generated on Caucasians. So we wanted to see how much these are conserved across the 3 major ethnic groups, the Caucasians, Mongoloids, and Africans. While many of them are shared, there are also quite a large number of methylation variants that are not shared. At the moment, I feel that it is mainly because of lack of data from other ethnic groups, but probably it is only a matter of time that we'll find more and more shared data. But whatever I'm showing, I think I'm trying to support my earlier statement that a life science researcher needs to know all the science areas. Only then you can do meaningful science. So this is a statistical analysis which is called as PCA. So it clearly segregated them into 3 groups based on the data. Right? The Caucasians is a distinct group, and then the Africans and the Mongoloids. And Indian data is very less, but they are more closer to the Caucasians, and these are the Spanish. Indian is a separate debate. I will skip that for now. And whatever I showed in the previous slide was all blood data, because blood is the most frequently studied sample, so you will have a lot of data on that. This is an example of a gene which is showing methylation variants across the ethnic groups. And here also you can see the distributions, like you have a bunch of samples with high levels of methylation and another bunch with low levels. And that kind of trend continues across regardless what kind of ethnic groups they belong to. Okay, these are all coming from different tissues, different people and so on. How would I know that these are heritable or not? If it is heritable, I should see it in all tissues from a single individual. And if it's not, the blank is filled by itself, right? So that again is based on limited sets of data. There are 4 cadaver samples and 6 adults, different types of tissues, but we have a handle that you can study different tissues from the same individual. So here the data suggests that the methylation variant number differs from the tissue type, which gives a first indication that they are not going to be uniformly distributed across tissues, whether it is cadaver samples or the adults. In case of adults, there are different types of tissues, buccal, hair and saliva, but because these are dead individuals, you can also take data from internal organs. So overall, 95 to 97% of the methylation variants are all tissue specific. Now here I take a pause and now turn to genetic diagnostics. So most of the time, epigenetic based diagnosis is based on blood. Now here the data is that if these are going to be tissue specific, you are studying the wrong tissue. No tissue is right. That's another thing. Very few, less than 5% are all systemic. Systemic means present in all different tissues. So that's one take home message from here, and you can see this is the same individual here. For example, for this gene, you have a different kinds of tissues, and this is how the patterns are. The same gene with different kinds of variation, and here the same individual, different tissues, right? So you see in some tissues it has less methylation, in some tissues it's higher, and these are data from across different peoples for the same gene. So how do we address reprogramming? You can do that in mouse, you can do all these things, but humans, you can't do. So one way is to look at induced pluripotent stem cells, where you can reprogram. You take a differentiated cell like skin or blood, any terminally differentiated tissue, and then you transfer it with 4 reprogramming constructs and you get a reprogramming cell, which is a pluripotent stem cell, behaves as if it is an embryonic stem cell capable of producing all different kinds of cell types. So if you look at the data now, fortunately again, there are 6 embryonic tissues, kidney, pancreas, lung, muscle, brain and skin, from which there are 2 independent pluripotent stem cell lines derived. And if we see the methylation distribution of these variants, they clearly segregate. All pluripotent stem cells are on one side and the tissues on the other side. And if you look at this more deeply, I have highlighted, I'll take example of only 2 tissues, kidney and muscle. Both are mesodermal origin, but you can see if they are mesodermal origin, you should expect them to be close. I should have expected muscle next to the pancreas or lung, right? But I don't see that. The kidney is far separated from the muscle, which means that even within the same individual, kidney is having different methylation variants, muscle is having different methylation variants. Therefore, the origins would have been after fertilization, after implantation, at later stages of development, probably as late as terminal differentiation. So, they are definitely not heritable. They are acquired. They are acquired probably stochastically because we don't know what the mechanism is. And therefore, even if there are organs derived from the same germ layer, they still can be different in terms of their epigenetic origins. This again is just substantiating the data. Earlier I showed you how the grouping happens. The PCA can also be used to identify what is the extent of diversity. Like for example, this is the data that is coming from the somatic tissues. Those 6 tissues which I have told before, like the liver, kidney and so on. And these are the pluripotent stem cells. So if you compare the width, the epigenetic variation that you see in the tissues is much more compared to the reprogrammed cell types. And this again, I'll just take the first example. If you see this, in one tissue, for this gene in an individual, you have one which is hypomethylated, decreased methylation, and a group of tissues which have increased methylation. So this is definitely a tissue-specific methylation variant. But after reprogramming, you'll see that this one has increased its methylation and gone up. Same thing you will see in other things. Some cases, it gets a loss of methylation. In some cases, it's a gain of methylation. But at the end of reprogramming, the main message is that whatever methylation variants that you have between tissues within an individual, all of them are significantly reduced after reprogramming. You can't address reprogramming in humans so easily, which I have told you before, but there is a handle to that. The sperm and the oocytes. You can't depend on females because they release only one egg at a time. Okay. And it is unethical to sample, right? So, sperm, there is no shortage of material, right? You can get a lot of sperm. So, males. So, you take the males and do the same thing, and we observed exactly the same pattern. In the sperm, the methylation variants reduce, and in the body fluids like saliva, blood, there is methylation variation. So what we believe is that reprogramming is a handle where when the sperm is going for the next generation and presumably the oocyte, they all start again with a clean slate. And sometime after fertilization, after implantation, and maybe as late as terminal differentiation, these methylation variants originate. And that makes each individual different from the other. So fortunately, it looks like they are not heritable. What does this mean? I have earlier said that these genes are associated with either mental health or cancer disorders. So we took data on 547 controls, prefrontal cortex, and 321 schizophrenia patients. And what we observed is, if you take the methylation variants between the two groups, they are almost similar. There is no real statistical difference. But, you know, genetics people don't get discouraged so easily. So, you turn now to, okay, these are the genes, but what if I look at only those genes that have been identified to be causing schizophrenia? There the number changes. There's a significantly high proportion of, let me show that if I have, yes. There is a significantly high proportion of schizophrenia associated genes showing methylation variants in controls, not patients. And we got these genes from a comprehensive study, again from our group. There are around close to 12,000 genes that have been implicated in schizophrenia so far. There is another debate, which again is my view, that what gene actually causes schizophrenia, because this 12,000 doesn't look far away from the gene number of humans. So presumably everything causes schizophrenia. But if you look at the controls in patients, you have a significantly high number within those schizophrenia group. And this is the Venn diagram. So a total of around 900 here among controls and just around 500 in case of patients. What we believe is, in genetic variation, higher the genetic variation, the population is better fit to face natural selection. So that's why large populations are favored, because they don't go extinct so easily. Small populations are more vulnerable. On the same way, what we hypothesized is that the complex disorder associated genes with methylation variants at a higher frequency than patients make the normal people better fit, and therefore they don't get disease in comparison to the patients. So just like genetic variation, the epigenetic variation also plays a role, probably, in natural selection. So nevertheless, this data is common between the patients and controls. So we took these 294 and looked at them further. And these are some examples of those schizophrenia associated genes. And you see that they're all distributed across different values. And so are their transcript levels. So of the patients and controls that I said, there is data that can be still tracked for about 100 normal people and 100 people on which we have methylation data and transcriptome data. So here also we see variation in the transcript levels that is encouraging. And these are some examples, right? These are some of the examples of schizophrenia associated genes where there is a significant effect on transcript levels, which means that these are schizophrenia candidate genes with methylation variation among normal people, and a subset of people who have this difference in methylation level will also have different levels of expression. That seems to be significant. How many of them? I'll tell you, not all of them. This data and a lot of other data which is not given here gives an estimate of something like 5 to 25% of the genes seem to be affected by the presence of these variants. The same thing we have observed in oral tissues, because as I told before, we don't really work on the pathways known, we try to find the pathways. So as part of that exercise, we have been working for some time on a precancerous condition which is called oral leukoplakia that causes an increased susceptibility to develop oral cancer. These individuals are more susceptible, that means they have a higher chance of getting oral cancer, but that doesn't mean that this individual will get cancer. I'm only telling that there is a higher chance, right? So we have that data, fortunately, this is our own. And here also there are 62 genes with methylation variants, of which 30 showed negative correlation and 32 showed positive correlation. There's much more data which I wish I could present, but I think the summary is clear that about as high as 25% of genes with methylation variants seem to be affected because of an altered methylation level, and that results in a significantly altered transcript level. At the time when we began, we wanted to see how much it translates to Indian, because Indians, we don't have any data. So we used a simple technique again, where we can measure the level of methylation in a sequence of interest. Again, I won't go into details of this because it's too technical. And we chose a couple of genes. One is called as NAPRT1, which is associated with schizophrenia, specifically in the South Indian patients. And this gene, out of 30 people, 5 have a variant. 5 have an increased methylation and 25 seem to have normal. And these 5 individuals show decreased transcript levels. That means these are normal people with no mutation, but their promoter is methylated, and therefore they are functionally heterozygous for this locus. This escapes conventional genetic diagnosis. Same way, there is another gene which we chose, which is called as AURKC. This causes an autosomal recessive male infertility. And here also, it's more widespread. Out of 30 people, 12 are moderately methylated and 7 are methylated. And again, these individuals produce less amount of transcript, which means lesser amount of protein. And even if they are genotypically same and normal, functionally they are heterozygous. And therefore, just I'll take a side step. If you have a situation like this, it is known in case of retinoblastoma, some rare cases of cancer are inherited. So in retinoblastoma, it's a tumor suppressor protein. There is a epigenetic mark that runs in families. These parents have increased methylation of one of the two alleles. So that methylated allele is not expressed. That means they are depending only on that one normal allele which is not methylated. And if that allele gets mutation, these individuals develop retinoblastoma. It has been demonstrated in families. Same way here also, you have two individuals genetically the same, epigenetically different, and therefore difference in susceptibility to disease. The only good news is that it doesn't look like heritable. So in conclusion, methylation variants are enriched in genes associated with certain complex disorders like schizophrenia and cancers. And a vast majority of them are tissue specific and they seem to be sensitive to reprogramming events. So reprogramming like gametogenesis helps in reducing this epigenetic diversity so that there is a reduced population risk for developing these disorders. And these in candidate genes, a subset of them show significant differences in their occurrence and controls in patients, of which another subset show decreased transcript levels, and therefore these are functionally different. And I have just listed some examples here. And therefore, since methylation variants are shared across major ethnic groups, there doesn't seem to be recent evolution or appearance of these things. And these are similar to the genetic differences that have been documented in literature. So with this, I will conclude this presentation. So you might be wondering from where we all got this data. I'll tell you, more than 90% or 95% of the data is actually mined from publicly available databases. So what I want to give a message to the young people is that our resources may be limited, but not our mind. So you can, if you have an original idea, you just need to determine yourself that you are going to do it no matter what. And there is enough of data available. Even if it is generated for a particular purpose, you can use it for some other purpose which is driven by your own idea. So Lav Kumar is a computer science faculty who helped us with a lot of Python scripts. I also learned some Python out of necessity. But as long as I feel I am a student, I am fine. I can learn more things. So he is mainly responsible for us to be able to analyze all this 3,000 plus epigenomes. And my colleagues, whom I bother them with my own research and that kind of things, coffee table talks like Trinad, Amartya and so on. And our director, this is another example I want to give you. He is actually chemist. He's from chemistry, IIT Kanpur. But you can straight talk to him, any biology research. And he will answer technically. And of course, our staff of biological sciences, our institute, because they have provided a very nice dedicated network facility so that we could analyze all this data. And including my institute and all other funding agencies which supported our work in one way or the other. And one of the most important person whom I want to thank in this is this exposure to the field of epigenetics. And he's my supervisor, Professor Sharit Chandra, and his thing is that he doesn't discuss with his students any of his bench work or lab work. He's a theoretical biologist, but believe me, I think I got this habit of thinking myself independently from him. And there is no limit to what we can. Some can be addressed today, some will be addressed tomorrow or later at some point. So with this, I end my talk, and I hope I'm on time and ready to take questions. And with a caution, as I began saying that I don't know, that's why I'm presenting.
I have questions. For you.
Take the mic.
I take mic for myself. What an outstanding talk. I really thoroughly enjoyed it. Yeah, so I was about to ask you the mechanistic difference between the individuals for the MEVAs, but we said we don't know the mechanism.
No, probably we don't know that is the right answer. But when we looked at more in the databases like the ENCODE and the epigenome roadmap, these genes are enriched with H3K27 triangulation and the rest. Now, the ChIP-seq data predictions seem to be associated with EZH2 major and also SVC12. Then the other I told you, rest is much less significant but still significant after the bond connection. But interestingly, rest is also a repressor mode.
No, my point is actually like, is there any enhancement in the DNA methylase activity in those individuals specific to those promoters? If so, how is that achieved? It has to be some
how can it start?
Exactly, in different individuals.
Yeah, I think we can talk about it because the thing is, we have observed that DNMT1 levels, we haven't looked at DNMT3A and 3B. Among normal people also, there is a difference in the level of transcripts between, just a separate work, but DNMT1 levels are also different between people. But DNMT1 is a maintenance methylase. You need somewhere a de novo methyl transferase to establish these things. So the Waddington's hypothesis of driving different cell types is through acquisition of random epigenetic marks, which many of them meet with a dead end, but some of them survive and make the tissues that we have now. So I attribute to these as more stochastic processes, which have sustained life, and therefore they have contributed to these organs and tissues.
So I have two more questions. I'm sorry, I'll take five minutes.
No worries.
It's very interesting. But we can also discuss outside. I limit my questions to 2 or 3 minutes. So another thing is like, is there any purpose of this diversified methylation among individuals? And even if you talk about one individual, you say tissue to tissue, there is some more methylation variation. Is it just a kind of random event that is happening without any consequence, or is there a consequence associated with it?
In a majority of them, it is not causing any transcript level difference, right? It is at best something like 30%. Now, why we have methylation variants in people is, as I told you, I feel that the increased epigenetic diversity makes this population fit for selection. Even if it is getting wiped out through reprogramming, they have to still survive first to transmit their genetic material to the next generation. So I feel that the stark difference between the controls and patients, I expected to have patients with more methylation variants, but it was the other way. And that's what the science throws up these challenges, which made me to rethink, and I feel that the better epigenetic diversity that we are seeing in controls is the reason why they are controls, and therefore better fit. That's a hypothesis to test.
So I will ask one last question. Remaining we'll talk during lunchtime. If, hypothetically, if I have 2 inbred population of mice and grow them in different environments, do you still expect to have this kind of diversified methylations? Or is that same loss cycle?
Yes.
You have fantastic information.
Thank
you. Audible? Yeah. I mean, you may find my questions maybe stupid, but I wanted to ask. But I have 2 questions. Question number 1. You mentioned that certain genes got replanted, and in human population we have several copies of the same gene. So all the copies of the genes get methylate at the same position. That is my question number 1. Question number 2. there are set of genes that are connected with a specific pathway, for example, JAK-STAT pathway, any other pathway. Is there any consensus way of getting methylated? The set of genes that are involved in a specific pathway, is it identified so far?
First, let me ask the second question. There are some specific pathways which have been significantly enriched, means multiple genes involved in the pathway affected. So over and above the standard significance calculations, we also impose bond-finite interactions to ensure that we get the most significant possible pathways. So there's a list of pathways which seem to correlate well with the disease as well as the protein within infections. Now coming to the first question, the very origins of different family members within a gene family is to ensure that these genes function similar but different ways. So by saying so, HOXA mutations, for example, they are associated with a specific type of disorder, but not HOXA. So even if it is a family member who is, let us say, having a methylation variant, and even say that that particular variant causes a difference in gene expression, would still have a different kind of outcome. Whether it contributes to susceptibility to disease or not needs to be tested. Probably we need to have much more larger data on both single gene as well as complex disorders. But we are looking at it. We have a collection of schizophrenia patients. If our theory is right, NAPRD1 methylation should be more represented in control sub patients. I mean, it justifies our statement either ways. Because if it is more in controls, which means that it is in support of our notion that the increased epigenetic variation favors that particular group of population. If not, because it's known this is susceptibility gene, it's straightforward that it's a candidate gene which is hypermethylated and you have significantly higher number of patients and therefore it explains as a risk. So there are explanations, there are unique positions in biology especially where you can have logical explanations for both kinds of outcomes. This is one such case. Okay, I wish to thank.
I have one. Actually, we use some machine learning algorithms for the prediction of diseases. Is there any algorithm that suggests cures?
Is there any algorithm that suggests
Cure. Remedy for the disease. Now we have now for the prediction.
It's a difficult question to answer. While I would say that the possibility is not zero, it's going to be extremely hard. I am only not so optimistic about it, not because I am not a computer scientist, but because there are many single gene disorders for which the mutations are already known. The candidate genes are also known. And it has been decades since we know that. And so far, even for a specific thing, we are not able to come up with a clear cure. Except for CML case where there is a mutation, there is a fusion gene, fusion protein, which is unique, not unique. So you can predict many things, but how much it translates to the cure, not let alone clinical trials or treatment or anything, how much it guarantees an individual to be walking out of the clinic cured of the disease. We don't have more than 2 or 3 examples.
Okay.
But actually, if you know the cause, you have a hint for the cure. So to fix the cause of the mutation, you develop the cure. Developing the cure is actually a problem. That's what it says.
Any questions? No.
Okay, I'm sorry for the students in case if it has gone overboard and too technical, but thank you then.
Thank you, sir. Thank you so much.
Thank you, Dr. Nagamohan Pama, for elaborating human disease susceptibility, methylation, DNA methylation dynamics, consequences, and genetic disorders. So your session is elaborative and informative. I hope this session will helps better for both student and faculty community. Thank you all for giving me this opportunity.
Thank you so much, sir. Now we have a small gratitude from our college. We want to felicitate Sir Nagmohan Garu by our department members, and Babu Rosas wants to join with us.
You have to, you cannot escape that. You cannot escape.
చూపిస్తుంది
It's not difficult.
సార్ మరి చాలా ట్రైనింగ్ లో ఉన్నారా
మీరు స్టేషన్
ఎందుకు సార్ చేసేవాళ్ళు అదే సరే తర్వాత పెట్టేది ఉండేది
రెడీ అయింది మేడం గారు ఉదయశ్రీ గారు ఇప్పుడు చూడండి సార్
ఇన్వైట్ చేయాలా
ఉదయశ్రీ గారు కదా మేడం ఇన్వైట్ చేస్తారు సార్ ఇంట్రాక్షన్ చేస్తారు కదా
డిసైడ్ చేసింది అంటే అప్పుడు
రమణ గారు ఆ మైక్ ఎక్కడ
Yes, I do that. Then I last person. But these three, I perform one also there is no guarantee. So we have to get cases. See, I think better I will send her. Yes, I will. Yeah. Yeah. Yes. Yeah, yeah. That's fine. That's fine. Yeah. But don't worry about it. I'll contact you also. But let my son come. No, we can go. How about your kids? Your kids are there only.
Hello.
Right now, I'll send the details of
Hello. Check. 1, 2.
Right now,
Yes, good afternoon. But are you out of the lunch mode? Are you still in sleep mode now? Just activate for some time. We will be having most informative session now. Yes, okay. Then this is Dr. K. Udayasri, HOD, Department of Data Science and Artificial Intelligence. It is my great privilege to introduce Dr. Kishore J.R. Konapalli, an eminent scientist in the field of immunology and cell therapy. Dr. Kishore earned his PhD in mechanisms of antigen presentation from the prestigious German Cancer Research Center, DKFJ, where he graduated magna cum laude. His academic journey also includes an MTech in biotechnology from Jadavpur University and MSc in microbiology from Acharya Nagarjuna University. Dr. Kishore had an illustrious career spanning academia, industry, and transitional research. He has held key positions as Chief Scientific Officer at Nexogenics Life Sciences, Vice President at Ortrin Biotech, and Research Scientist at Singapore Immunology Network, where he made significant contributions to the understanding of lipid antigen specific T cell responses. Currently, Dr. Kishore serves as a consultant at Pulse Pharmaceuticals, where he is spearheading cutting-edge work in CAR T cell therapy, guide and tried molecules, and SCFP drug conjugates. Under his leadership, Pulse Pharma has established a state-of-art cell therapy division in Hyderabad, focusing on affordable and scalable cell therapy solutions for oncology, autoimmune disorders, and infectious diseases. His expertise spans immuno-oncology, CAR vector engineering, protein purification, nanoformulations, gene delivery systems, and clinical assay development. Beyond his scientific achievements, Dr. Kishore has also contributed to entrepreneurship, having co-founded Nexogenics Life Sciences, a diagnostic company that successfully developed ELISA kits and rapid test systems under WHO GMP standards. He has also been certified the FDA Clinical Investigator Training Course and holds international accreditations in animal research ethics and laboratory practices. With his wealth of knowledge and pioneering contributions to the development of next generation therapies, Dr. Kishore continues to inspire innovation in biotechnology and clinical research. Please join me in welcoming Dr. Kishore J. R. Konpalli to give his lecture. Thank you, sir.
Hello, can you hear me? So good afternoon. So after having lunch, everybody is sleepy. I want to talk to the students, not with the front row people. Second row, so faculty. Right. Now association with Siddhartha College at first. 1995, MSc Microbiology అయిపోయిన తర్వాత I got selected as a lecturer in this college, taking care of BSc Microbiology. I don't know how many of you heard Mumuni Subbarao garu. ఆయన చేతుల మీదుగా నేను అపాయింట్మెంట్ ఆర్డర్ తీసుకున్నాను. I got already a third rank on the entrance test. That's why I left Siddhartha to pursue my MTech. So నా వల్ల చాలా ఇక్కడ పార్ట్ టైం లెక్చరర్స్ బ్యాడ్ జరిగింది ఆ రోజుల్లో. ఎందుకంటే నేను వెళ్ళిన వెంటనే మిగతా పార్ట్ టైం లెక్చరర్స్ అందరి దగ్గర సర్టిఫికేట్స్ తీసుకున్నాను. వాళ్ళు నాకు ఫోన్ చేసి తిట్టారు. Okay, that was a funny incident. Anyway, see, మీరు బిఎస్సి స్టూడెంట్స్, నేను చెప్పేది మీకు కొంచెం హెడ్ ఆఫ్ ది మీకు అబౌవ్కి వెళ్ళిపోతుంది. I will try my level best to give my lecture as low as possible. Okay? Okay. Yeah, these are all. Can you hear me now? Okay, these are all my affiliations. Like, I did what. Currently, I am located in Pulse Pharma as a chief scientific officer. And my title of the talk is Treat the Cancer with Self, not with the pill. Right? Cancer patients need, అసలు ఎవరైనా చూసారా స్టూడెంట్స్, కెన్ యూ లిఫ్ట్ దేర్ హ్యాండ్స్? మీ ఫ్యామిలీలో కానీ ఎక్స్టెండెడ్ ఫ్యామిలీలో కానీ అసలు ఓకే అట్లీస్ట్ ఒక 10 మెంబర్స్ కనపడుతున్నారు. వాళ్ళు ఫేస్ చేసే ప్రాబ్లమ్స్ ఇంత కాదండి, అది కేవలం చూసిన వాళ్ళకి మాత్రమే తెలుసు. సో ఇట్ ఈస్ a kind of very difficult to deal with it. Now, yeah, I wanted to introduce my director of the institute detailed, who got Nobel Prize in 2008. ఆయన HPV అనే వైరస్ని ఆయన ఐడెంటిఫై చేశారు. He shared Nobel Prize with Montaner and other scientists who identified HIV. And don't get surprised, this is me. In 2006, I was looking like that. So he's my boss. Okay. So this is how they celebrate. When I just got passed out, when I just defended my PhD thesis. Okay. Okay, I don't want to give the business lectures here, so I'm skipping all these things. Okay. I'm going to talk about CAR T-cell therapy. First of all, మీలో ఎంతమందికి T-cell అని పేరు ఉన్నారు? Almost all? You know what is T-cell, right? Okay. So we have lymphocytes. T-cell belongs to the family of lymphocytes. Why we need to study more regarding the T-cell is that it is the most potent cell. In our body, only 3 cell types can capable of killing the other cell. One is T cell, another one is NK cell, another one is NKT cell. Okay. So T cell, like B cell, they have their own receptor. You know, that's called T cell receptor. In the same way, B cells also have B cell receptor. So with receptor, it is supposed to recognize the antigen, right? So T cell receptor, through receptor, it is supposed to recognize the T cell antigen, which is presented by something else. Don't much worry about that. The problem comes with the cancer patients is that if your T cell is really working well, why should we get the cancer? That is the first question. If your immune system is so capable of attacking, మీకు ఫీవర్ వస్తుంది తగ్గిపోతుంది కదా, రైట్? మీకు ఏదో మలేరియా జ్వరం వస్తుంది తగ్గిపోతుంది, ట్యూబర్క్లోసిస్ వస్తుంది, అది కూడా మీరు మందు వాడికి తగ్గిపోతుంది. బట్ క్యాన్సర్ ఎందుకు తగ్గడం లేదు? దట్ ఈస్ ద ఫండమెంటల్ క్వశ్చన్. సో క్యాన్సర్ సెల్స్ somehow they're actually, immune system ని పడుకోబెడుతుంది. Mechanisms of just try to understand the concept, right? మన ఇమ్యూన్ సిస్టమ్ దాన్ని అటాక్ చేయలేకపోతుంది, అందుకే మీకు క్యాన్సర్ వస్తుంది. ఓకే. ఇప్పుడు ఈ CAR T-cell థెరపీలో how we overcome that problem, in what way it is better than the original T-cell. మీకు ఫార్మా కంపెనీలు ఎవరైనా విజిట్ చేశారా? ఎనీ మ్యానుఫ్యాక్చరింగ్ యూనిట్ ఒక అమ్మాయి చేయలేదు. ఓకే. ఫార్మా కంపెనీస్లో ఒక పెద్ద వెసల్లో ఒక బ్యాచ్ వేస్తారు. దాని నుంచి ఒక 10 లక్షల టాబ్లెట్లు తెప్పిస్తారు. అంటే ఒక బ్యాచ్లో 10 lakhs టాబ్లెట్స్. దట్ ఇస్ ద నార్మల్ వే ఆఫ్ బ్యాచ్ మ్యానుఫ్యాక్చరింగ్ సిస్టమ్. కానీ సెల్ థెరపీలో we take out blood from the cancer patient. We do something with the blood. And then, upon modification in vitro, we put back the same blood into the same patient. In scientific terminology, we call it as autologous, infusion. Okay, blood తీసుకుని patient నుంచి do something and put back into the same patient. Okay? What we do అనేది I'll explain in the other slides. So the summary of this slide is that in one batch, in a normal conventional drug manufacturing, you can attend lakhs of patients, whereas in cell therapy, the starting material, raw material, and caters to only one patient. That is the main difference. That's why it is very expensive. Okay. In this slide, I will explain how CAR T-cell therapy works. See, from the patient, we draw the blood, and in the blood, from the blood, we will isolate T cells. There are several mechanisms to isolate T cells. And then we insert a specific gene that is called CAR gene, chimeric antigen receptor gene. That CAR gene we will introduce into the T cell. Now that CAR gene get expressed a protein called CAR protein. As you see here, chimeric antigen receptor protein. This protein is specific to bind on a protein on a cancer cell, which means now the T cell, normal T cell converted to CAR T cell, the CAR receptor is specific to identify and bind a specific protein on the cancer cell, thereby it can recognize. We expand them, these CAR T cells in vitro, into the millions of the cells, and then we re-infuse back into the same patient from where we got the blood. Okay. Now the patient is having cancerous cells and the CAR T cells together. Now CAR T cells can fight with the cancer cell inside the patient body and kills the cancer cell, so that you can see the eradication of the cancer. This is a very simple, looks like very simple procedure, but it has a lot of complications, which I don't want to discuss with you. Okay. Hope you understood the concept. Okay. See, this is a patient photograph. You see all these things are, this patient is basically suffering from diffuse large B-cell lymphoma. And this is one of the clinical trial data that I'm showing you. And see, this graph, the graph showing you after 10 days of the CAR T cell infusion. Okay. In the previous slide, I have shown you how to prepare a CAR T cell. In that way, they prepared CAR T cells for this patient also, and they infused it back into the same patient. After 10 days, the photograph looked like this. That is the leg of the patient. See, at day 50, you see complete killing. Okay? And if you look at the day 330, it looks like completely normal line. Okay? As if there is no cancer at all. That is the beauty of the CAR T cell. On an average, I'm summarizing some of the clinical data. On an average, within 3 weeks, 90% of the cancer burden is gone. This treatment. This treatment in US, it is costing 6 crores in Indian rupees. We are working to bring down the cost to 50 lakhs. That's what we are working with. This is another example of the patient, a female patient suffering from multiple myeloma. Okay. We have B cells and T cells and lymphocytes. And once the B cell terminally differentiate, you can see the plasma cell formation. Plasma cells are the cells that produce the antibody in our body. If the plasma cell become cancerous, you can say multiple myeloma. Okay. See, this is just before the treatment of the CAR T cell therapy. And after 6 weeks, all these black dots, this is the PET scan. All these black dots belongs to the kind of, there is one dye, FDG. All fast growing cells take up this dye. That's why you can see in the PET scan, black spots. All these are nothing but representative of your tumor cells. After 6 weeks of CAR T cell infusion, you see 90% of. So the beauty of the technique is really very high. Now I come back to the original first discussion. What I said at the beginning, if your immune system is so good, there should not be any cancer at all in the first place, right? Now, how you overcome the normal classical immune pathways? I think it will go beyond your head, but I explain. There is one molecule called MHC, major histocompatibility complex. If anybody wants to donate a liver or kidney, something like that, you might have heard HLA matching, HLA tissue type, right? It's a scale of 10. Minimum 6 or 7, it should get matched. Then only a donor can donate his organ to the recipient. So MHC is nothing but, major histocompatibility complex is nothing but a tissue typing parameter. Okay, which means if the MHC is not matched, the recipient organ will be rejected and the organ will be attacked by the recipient immune system. In case of identical twins, where you have 100% genetic match, right? They can donate in either because they are 100% genetic matched people. Whereas that's not the case with unidentical twins. Okay. Now the point is that all antigens that are to be presented to our immune system has to be presented by an MHC molecule. Okay. Which means if a cell transforms to a cancer cell, let's say, a cancerous protein that also has to be processed inside the cell, that also has to be presented by MHC to the immune system, right? If there is no MHC expression in the cancer cell, how cancer cell can present the antigen? It can't. So the first point is that a cell, a cancer cell, to establish its kingdom, the first thing is that it should shut down its MHC, so that it can escape from the immunity. Now, how can you overcome that MHC dependency? As I mentioned in the first slide, one of the first slides, the CAR gene. The CAR gene activates the T cell independent of MHC. Doesn't require MHC. So that you can see the activation of the T cell directly through CAR receptor, and thereby T cell gets activation. There is a pathway called perforin granzyme pathway that will get triggered, and through that pathway it will kill the target cancer cell. This is another example. This patient is suffering from glioblastoma, brain tumor. This is before operation for the brain tumor. You see here, the white mass is nothing but the glioblastomic mass. And this is after surgical resection of the tumor. You see there is no white mass here. And after infusion of the CAR T cells, you can see a complete regrowing the normal brain mass. Okay. So now there are, for several few cancer indications, we have CAR T cell therapy, but not for all. Okay. So how do you design a CAR receptor? It's a combination of the antibody it's a combination of antibody and T cell receptor. See, this is the antibody structure that binds to the antigen. So you take out this one, and you take the signaling domains of the T cell receptor here. This is called as CAR receptor. And as I mentioned, this is the tumor antigen on the tumor cell, and that will be detected by the CAR receptor, and it will targetedly kill the tumor cell. This is So how do you manufacture these CAR under GMP conditions? Okay. So in pharma grade, you have to have a GMP facility to manufacture the drug. Okay. So this is the machine that we normally use for GMP manufacturing of CAR cells. I don't want to discuss these things. Dr. Mohan? Mohan? I want your attention in this one. This is a CD19 antigen on the B cell. And there is a variant in the human population at 174th amino acid, we have leucine and a valine. The leucine is a kind of a white type one, which is having high affinity binding with the conventional CAR molecules. That was improved. Whereas, if it is valid at 174th amino acid, the improved CAR molecule is having low affinity. That's why you are getting some kind of relapse. So why all CAR-T treated patients are not completely, able to kill the cancer, right? This is one of the reasons. So we came up with a novel CAR construct in my company. The American companies targeted the distal region of the binding site for a CD19 molecule, whereas my company, we targeted the membrane proximal region. The reason for that is two things. The 174th amino acid will not come into the picture for this one, for our targeted epitope. So it doesn't matter whether it is leucine or valine. That is number one. Number two, in an immune synapse means when T cell meets the target cancer B cell. Okay, these two cells, they kiss each other. That kissing portion is called immune synapse. See, in every cell type in cell signaling pathways, which Dr. Shivakumar will explain in detail, there are some negative signals and there are some positive signals. Cell will summarize how many positive signals I am getting to expand clonally, how many negative signals I am getting to do the same. It will summarize whatever the result comes, it will apply. So there are some negative regulators like CD45, which comes into the area of the immune synapse, and thereby they negatively regulate, and thereby the clonal expansion will be impaired. If you target the proximal epitope, you can prevent the intrusion of CD45 into the synaptic area. That's how we can make it better. So this is the molecule that we designed with the CD19 single chain fragment variable SCFE, and it interacts with the extracellular domain of the CD19 antigen in a much representative manner. Here it looks like this is our molecule that we developed. So it is binding with the wild type and as well as with the delta 2 exon CD19. Okay, and thereby we can increase the efficacy. And this is a CD20. CD19, CD20, both molecules are expressed only by P cells, not on any other cell type. Okay. Now, so either by exploiting CD19 or by exploiting CD20, we can kill the target tumor cells. So we did, this is the antigen, CD20 antigen. This is the molecule that we designed in our company. And this is how they interact with each other with the extracellular domain. And we also designed a dual CAR molecule which binds simultaneously with CD20 antigen and as well as CD19 antigen. I better skip this part. Okay. That should one is also not in favor. Okay, in summary, in Path Pharma, we are coming up with 3 different products: CD19 CAR, CD20 CAR, and 19 and dual CAR. Okay. There is another way of activating T cell. Okay, how? Just look at this cartoon. TAA means tumor associated antigen. That antigen present on the tumor cell. And this is the T cell, and this is the T cell receptor. CD3 is part of the T cell receptor complex. Okay, and this molecule is called BiTE. BiTE's expansion is bispecific T cell engagement, which means it engages with one arm with CD3 on the T cell, and with the another arm it binds to the tumor associated antigen. Okay, it makes as a kind of a bridge between the target tumor cell and the T cell. So since it is bispecific, it is called bispecific T cell engagement. And once the engager engages these 2 cells receptors, you can see the activation of T cell, and thereby it can kill the target tumor cell. This is how the BiTE molecules work. The first BiTE molecule got approved in 14th June 24, very recently, from Amgen. It's called Blincyto. And this is the BiTE molecule that we designed in our company. So in a graphical cartoon manner, one arm binds with CD19 on the B cell surface, and with another arm it binds with the CD3. And this is how the molecule looks like. And we also developed a CD20 BiTE as well. And we came up with an idea of attaching a human serum albumin binding domain for our BiTE molecule, because the FDA approved BiTE molecule half-life is only, serum half-life is only 2 hours. You need to take, the patient needs to take 28 injections in a span of 56 days. Which means, why a molecule that was introduced intravenously having less half-life? Less half-life means it is not available in the blood anymore after introduction. That's why we call it as a kind of half-life. You know, how long it stays in the serum. If it is not staying in the serum, you can't see its effects, right? So, if you introduce a molecule, it should stay for a longer period so that it can act for a longer period, so that you don't need to introduce the same molecule by intravenously or any other manner that is recommended, so that you can minimize the side effects. So, to increase the serum half-life, we came up with an idea that we choose 2 different ways. One is, already we have majority of the serum protein is human serum albumin, HSA. That HSA binding peptide we attached, and we also made a fusion protein directly. The BiTE molecule and HSA fusion protein, so that the half-life of the HSA is 21 days. So we wanted to check in our clinical trial whether our bite molecule is also, there for 3 weeks or not. That we need to check in the preclinical data with the mice data. And like CD19, we also made a tri molecule with CD20 specific as well, and a pure specific, like a kind of quarter specific T cell engages as well. And there is one more molecule called PD1. I will make it very simple. What is PD1 for you guys? If your own T cell, if the cancer patient's own T cell has to attack the cancer cell, another obstacle is that there is a kind of the molecule by name PD1 available on the surface of the T cell. Whereas cancer cell expresses its ligand called PDL1. So the moment T cell try to scan the cancer cell, you will see PD1, PDL1 interaction, so that the cancer cell sends the signal to the T cell, I'm not your enemy, I'm your part of it. So don't kill me. That is the kind of signal T cell gets from the PD1, PDL1 axis. If you could break that axis, PD1, PDL1, the T cell empowered to recognize the target cancer cell in a more effective manner, and thereby the T cell can kill the cancer cell. There is one injection, a monoclonal antibody by name called Keytruda. That is, the annual sales of Keytruda is around $24 billion US. Okay, that actually disrupts the PD1, PDL1 axis. In a similar manner, if you can disrupt by using this desired domain, which acts as a kind of a blocker, so that you can also empower the T cell to get activate and kill the target tumor cell. So this is, we call it as a pentaspecific T cell engagement. So in our lab, the T cell engager platforms we built, we have 4 domains in my lab. Computational biology, where we do a lot of bioinformatics, and we have highly skilled genetic engineering technicians with us, and cell biology, we can call it as immunobiology, you can say, and we have highly skilled protein purification team with us, right? Okay. Now, I'll introduce another type of molecule called antibody drug conjugate. If you are suffering from fever, let's say, you're taking paracetamol, 650 milligrams, Dolo 650. It is very famous during the COVID. Everyone takes 650 milligrams. It is a highly liver toxic. Right? Assume that by taking just 1/100th of the amount, instead of 650, 6.5. Right? If your fever is gone for at least 6 hours, would it be not good? It is excellent, right? Which means, how can you reduce the drug concentration amount to be taken for its effect, right? So the point is that the cancer patients, they are taking a lot of chemotherapeutic drugs, and they suffer like anything. They lose all their hair, and all because the cocktail of cytotoxic drug molecules, it kills all phosphoryl cells because it doesn't discriminate which one is cancerous, which one is your bone marrow cell, which is one of the phosphoryl cell, your hair follicle, which is also a phosphoryl cell. It kills everything, all phosphoryl cells. Okay, that's why they listed. What would be the situation if the amount of chemotherapeutic drug that are normally administered in chemotherapeutic procedures, if it is 100% amount only, that is fantastic, right? That is the part of this ADC concept, antibody drug conjugate. How the chemotherapeutic drug so this is the antibody. This antibody, you know, they tagged with a cytolytic drug. For example, toxophilus, one of these cytolytic drugs. Okay, and this antibody specifically binds with the molecules that are specifically available on the cancer cells per se, and they bind. They form a complex like this. Once antibody bound to a cell surface antigen, you can see the internalization of the complex. And now, along with the antibody, the drug is also entering into the cell. And once the complex forms a vesicle called endosome, it eventually fuses with the lysosomal compartment. Okay? You know, lysosome pH is very low, around 4, whereas endosome pH is around 6.5. When they start fusing with each other, you will see a kind of pH 5.5 or something like that. But immediately the proton pumps that are available in the endolysosomal compartment membrane, they start kicking on and they start reducing the pH, and they will bring back pH to around 2.5 again. During this process of the bringing down the pH, you will see a lot of proteases that are actually getting active at different pHs. Okay, so cathepsins are one of the class of proteases that are available in this complex. Those cathepsin molecules, this is the drug molecule which is attached with a linker to the antibody. This linker can be cleaved with active cathepsin, so that in the endolysosomal compartment, you can see the release of these drug molecules due to the cleavage, and those drug molecules enters into the nucleus of the cell, and thereby it kills. Which means 1,000 concentration of the chemotherapeutic drug can kill a single cell with a very, very minimal amount of the drug. Okay, so you can reduce almost no side effects, almost. So we are coming up with 3 different SAP drug conjugates: CD19 based, CD20 targeted, and VR based.
early career research by 4.45 lakhs. And the second one, he completed Welcome Trust DBT Fellowship with 3.5 crore. And presently, he is another SERP project is going on there.
Sir.
And he published around 15 articles with 5,751 citations. So we want to give a good clap because with a short number of articles, he got that more than 5,000 citations. So I am getting jealousy on him now. As a doctorate, I am also, but I am unable, even though I published around 60 publications in SCI, but I was unable to get that number of citations. So sorry, sir, I am very jealous to you. Because with the number of a few publications, the 5,751 publications is not a joke. It is very tremendous, and it shows that the importance of the publication, how much you have the depth in the publication. Otherwise, no one cannot recite that paper. So, see that within 15 publications, he got 5,750 publications. So, we want to give a good clap in our presence. So, I hope that you will enjoy the section Transcriptional Respiratory by NFKB in Cancer. So, I think there's a total research area is in cancer, I think in cancer. So, he's a master, he's a postdoctoral, and he's a doctor in philosophy. He's totally running in biological sciences with cell signaling, gene regulation, immune system, cancer biology, and molecular biology. So, on behalf of our biology department, once again, I am inviting to motivate with its knowledge to our students. Thank you.
I really thank him a lot for all the kind words that he spoke about me. And also thank Siddhartha College for inviting all of us here and giving us an opportunity to interact with people, to share our knowledge and try and motivate the young minds as much as possible. So, and for all the hospitality that we enjoyed so far. I do not want to straight away go into a lot of details about science, molecular biology, signal transduction, pathways, gene regulation, all that is happening right now in all of us. If you are sitting here and listening to me, there is some process going on, and that is regulated by your genes and proteins right now. And likewise, if the function of those genes is kind of messed up, like Professor Nagmohan wrote, methylation errors and all mutations and so on, that would mess up the whole system. You end up with having cancer, immunological disorders, developmental disorders, and so on. But before I go into those kind of details, you have been listening to a hell lot of you have been going through a hell lot of aspects about microbiology from Professor Lancharaya, DNA methylation and gene mutations and so on from Professor Nagmohan, and immune therapy from Professor Kishore. I will go into science a little bit later, but let me ask you a question. How many of you have, listen to me carefully, it will be fun talk actually. How many of you have failed in 10th class or +2? Students or faculty also. How many of you have failed in 10th class or +2? Students and faculty, both can answer. No, there's no problem. Fantastic. Anybody else? Great. I failed in +2.
I failed in +2. +2 chemistry.
So I believe in the statement, whoever made, I don't know, failure is the first step of victory. You fail, you learn from it. Okay? So now I tell you some more interesting aspects, okay? When he fails in +2, naturally, my brother is very intelligent guy. I am jealous of him. He always succeeded in his +2, BSc, MSc, and he ended up joining Indian Institute of Science, from where Professor Naramohan has done his PhD. Indian Institute of Science, Bangalore, is the ultimate place in India for doing science. It's very difficult those days to get a PhD position in IISc Bangalore. My brother was a PhD from there. At that time, I failed in first. So you can imagine my situation in the family. So everybody used to started saying that I'm a useless fellow. You failed in +2. You can fit for nothing kind of a guy. And my brother, you cannot say, he always used to insult me that you are hopeless guy like that. So what I'm trying to say is that I started my academic journey by being labeled as a hopeless, useless fellow. Okay, that was my first step. Then I had to prove somehow, no, I'm not hopeless. I can do some work. At least all of us should be able to do something in our life. So I had to prove that I am not useless. So I worked hard a little bit. And somehow I got into BSc in Hindu College and completed BSc somehow. And then to really prove that I can do something, I can achieve something in my life, I studied a little bit to get into biochemistry. Those days, believe me, now also, biochemistry is such a wonderful subject. I don't know why people are actually, I'm very sorry, I'm really disappointed to know that the master's program is terminated here. Because biochemistry is the stepping stone to understand your own biology. It's not something random stuff. Your own biology you have to learn by doing biochemistry. And see, if you want to do mathematics, physics, chemistry, or you want to earn money by doing real estate business, you have to be alive. For you to be alive, you should understand your own body, what is happening in your body, and what is how the mechanisms are regulated. For that purpose, you must study biochemistry. Anyway, to cut the long story short, I studied well enough to get selected into biochemistry program of Andhra University, and then I had trouble again. Because of my nature is that I am not a very dedicated student. Okay, I had my type of friends, not Lancharaya, he was my classmate. He is very, very disciplined and very hardworking student. So, no, it's the truth actually. Whatever I'm saying is not exaggeration, it is truth. He is very, very disciplined. Always used to go to library, study, and he got very good marks in his MSc biochemistry. He cleared.
Siva, you say hardworking.
Intelligent also. Because he cleared CSIR. You know, how many of you know what is CSIR? Council of Scientific and Industrial Research in India, that provides fellowship for students to do PhD. Back then, for the entire country, some 20-30 fellowships used to be given. Out of 20-30, from a university like Andhra University, where nobody knows anything, he cleared CSIR. That's kind of what the hell, right? So then, I was again negligent and very bad in learning. So I did not study well. Somehow I got a first-class degree in MSc Biochemistry, but we used to have something called Viva. After the exams are over, we used to get 2 external faculty from outside the university to conduct Viva to test our actual knowledge. Are you really fit to get a first-class degree in MSc in Biochemistry, or you just got the degree just like that, put on the neck? So for doing that, we got 2 eminent scientists from India, Professor Ram Sharma and Shastri. Both were from Indian Institute of Science, Bangalore. They happened to be the previous students of Andhra University. So our faculty invited them to conduct the Viva. And during the interacting session with Professor Ram Sharma and Shastri Garu, who were professors at Biochemistry Department of Indian Institute of Science, our faculty of Andhra University questioned them, why you are not helping? You are our alumni. Andhra University alumni. You are now in Bangalore. You are not helping our own students to get PhD admission in Bangalore. Why are we not helping our students? Okay. So that question was posed to them. It was embarrassing for Professor Ram Sharma and Shastri to answer that question. How can they say that your students are bad? That's why we're not taking those students, right? So they could not answer that question, and they kept quiet. And during the Viva, I entered into the Viva room. Rama Sharma Garu, he asked me, what do you know in biochemistry? I was kind of mumbling here and there, and he said, so what is the end product of glycolysis? Okay. I said, I don't know. I had a first class degree in biochemistry, MSc biochemistry. I don't even know what is the end product of glycolysis. Then he asked me to tell one reaction wherein ATP is used. I said DNA polymerization because I used to interact with him sometimes. So he's an intelligent guy and he knows what is DNA replication. So DNA polymerase uses ATP. Then he asked me, okay, TK, now you tell me a reaction where GTP is used, a metabolic reaction. I said DNA polymerase. There also GTP is used to introduce G into one nucleotide into the replicating DNA. Then what about TTP, ATP, everything? For everything, I said the only thing I know at the end of the day was DNA polymerase. That also because I interacted with non-capsid. Okay? No, it's clear. Then what happened? Everybody started laughing at me that this guy doesn't know. And Ram Sharma Garu and Shastri Garu had a wonderful opportunity to explain to my own professors, honorary professors, the reason why we don't select your students to do PhD at IIT Bangalore is this guy. You are giving first class MSc biochemistry first class degrees to such quality students. How do you expect us to take them for PhD? It's an insult, a slap on my face, and also slap on the face of the department. So again, I felt insulted, and sometimes getting insulted is also not bad. It will inspire you to kind of come back with vengeance. What the hell? I'm not that bad. I can do better. So like that, I realized, and I wanted to study. Okay, again, to prove myself. Then I asked one of my classmates, another guy called Raman. He is also a good student. He had this textbook of Strayer, biochemistry by Strayer. By the way, if any of you, after listening to all of us, get motivated to learn at least the ABCDs of molecular biology, read biochemistry by Strayer. Okay? So I wanted to study biochemistry by Strayer, that textbook. I went to Raman and asked me, hey, Raman, can you lend me that book? That guy said, Shiva, for you, a local BSc level guide is enough. You will not understand anything in Strayer biochemistry. So forget it. That's another insult. But inherently inside me, I always used to think that I am better than Mancharya. Okay, that also is inside me. But I have to prove it. I have to prove to the world that I can do better. It's academic, healthy competition. Okay, I respect his knowledge, I respect his achievements, but I have to prove that I am better than him. So I have to work hard. At some point of time in your life, you have to work hard to do something unique that only you can do. One more thing I'm telling you. So I completed that and eventually I got selected in IIT Bangalore. I kind of gave it back to my brother who insulted me. I said, I am also a IIT PhD now. Then I left IIT. I went to Germany. I did my PhD. Then I went to postdoc with a world legendary scientist, Professor Michael Kerin. Our sir just said that 5,000 citations is really great, but I would say it is nothing because my boss Michael Kerin has 3,50,000 citations for his work. Can you imagine that? Okay, 3,50,000 citations for his work. He should get Nobel Prize sometime soon. Okay, and he published, if you know about journals, Nature, Science, these are the Bibles, Bhagavad Gita, and the Qurans of any science subject, Nature, Science. In those journals, he published more than 300 papers. Can you imagine? So, with that kind of a scientist, I got an opportunity to do to work during postdoc. Okay, I went to his lab. Now, what happens is again my negligence way of under-discipline or indiscipline. So excuse me for my English if I'm wrong in English. Indiscipline, the behavior again started again in Michael Kerin's lab, where every minute is kind of so valuable. Okay, I wasted time, and somehow I was doing here and there work, and he was very angry on me. He gave me a task to do, and I did that. But then he thought I did not do, and eventually what happened, I cut the long story short. He said, if I do not see this result from you, whatever work has been given to me, by today evening 4 p.m., if I don't see the complete full-fledged result, you are out of my lab. I do not see your face from tomorrow morning. That was the warning given. But I did the work. He went and came back and said, even if you have done the work, if it is not up to my quality, then also you are out of my lab. So, this kind of challenges are required in life. So I did the work. I showed him. I said, it's not really less than your quality. I challenge you. How can you say it is less than your quality? I told this to, out of immaturity or confidence, whatever we call it, to Michael Kerin, who is there. We are tiny dot in front of him. So Michael laughed at me and said, Shiva, it's okay. You are not useless. You can continue in my lab. What I want to say, listen to me. Michael Karin is a legend. He said, Shiva, you are not useless. You can continue in my lab. From being called as a useless, hopeless guy after failure in the 12th, I developed to a level being called not useless by a legendary scientist like Michael Karin, who had 350,000 citations. Okay, so that's my case. The bottom line here is that those people who study well, like Nacharya, let them go to wherever. Those who are kind of failing to grow, take me as an inspiration. If I can do, you can do also. You understand? Ishwar also is like me. Okay, with that, I'll start doing some science. So, currently, by the way, I am Sivakumar. Sivakumar is my surname. I am at Indian Institute of Science, Education and Research, Tirupati. Professor Nagmohan this morning mentioned that for current day biology, doing biology itself is not enough. If you are good in biology, is not enough. You have to be very good in mathematics, physics, chemistry, and everything, computational aspects, everything. You have to be very good there, and then do biology. At the end of the day, we all have to do biology because we still do not know the ABCDs of molecular processes by which we age, we become old, and we all die. The bitter truth is that the death clock is ticking. Maybe you are late. I am earlier than you. But one day or the other day, we all have to reach there. The death clock is ticking. We have no idea what the hell is happening in our body right now. I lost my hair. You know, what the hell? How the hell I lost my hair without my knowledge? I could not stop it. How ridiculous is that something is bad is happening to your own body and you cannot do anything. You run behind real estate business to earn money. You are going to die anyway. What will you do with all that money? So therefore, there is a bigger need for intellectual minds to work in biological sciences, to really understand the whole process of human body, to improve human health. And in our Puranas, if I'm not exaggerating too much, it is described that you can actually live up to 1,000 years, waste molecule for 1,000 years, right? So we can actually understand the molecular biology behind aging and improve human lifespan as well. Okay. What we are doing currently, in the next 15-20 minutes, I summarize. Is to understand, to identify commonly operated survival mechanisms in cancer by different signaling pathways. So the major problem about cancer, if you talk about generalized thing, is that normal healthy cells have a lifespan. They will die after some time. Cancer cells do not die. They have mechanisms to escape death. Okay? So, if you understand the mechanism by which the cancer cells are escaping the death, then you can target those mechanisms to bring out a drug for cancer. Okay, so, and then the problem is, initially, the major problem with regards to development of a cancer drug, we don't have a cancer drug so far. Even immunotherapy is failing. Okay. Today, we don't have a drug to cure cancer. Why? Because I'll tell you, listen to me carefully, it's interesting actually. You have normal tissue. Unfortunately, in the tissue, all cells are equal at the beginning. Somehow, one of the cells get a mutation, an oncogenic hit, or oncogenic mutation we call. That cell become abnormal. Okay? Due to, in a tissue, we have all healthy cells. Unfortunately, in some individuals, one cell get a mutation for some reasons. And that cell become abnormal. Okay? So these are the abnormal cells. They grow slowly because only one mutation happened. It does not cause rapid division of the cells to proliferate extensively. That one cell starts becoming malignant, means cancerous, slowly divides. During the process, these cells acquire more mutations. One mutation, two mutations, three mutations like that. And each mutation is different. Each mutation is activating one pathway to enhance the proliferation and survival of cancer cells. The second mutation will activate another pathway for survival of the cells. A third mutation will activate a different pathway for the survival of the cells. Are you following? You have three ways of survival. mutation A, mutation B, or mutation C. 3 ways of survival. Okay, so like that, more and more mutations happens. You can see the color for the green, blue, and red, yellow, orange, and pink, and so on. So that means each of these cells, which have been developed from one single tissue, the cells got more and more mutations. So eventually, you have a mixture of cancer cells in one tissue, one patient's tissue. In a one patient's tissue, you have a mixture of cancer cells. This cell is dependent on one mutation downstream oncogenic pathway for survival. This cell depends on a different pathway for survival. This cell will depend on a different molecule for survival. Now you come with chemotherapy. Okay? Why chemotherapy is so toxic? Because you are adding, let us say, there is a molecule called NFKB. Some of these cells might be dependent on NFKB for survival. Some of these cells might be dependent on MAP kinase for survival, and some of them may be more dependent on AKT for survival. Okay, relative dependency. So you add an AKT inhibitor. These cells will die, but these cells will still remain. They will keep on growing because they depend more on other pathways, less on AKT pathway. You add AKT pathway and MAP kinase pathway, then these cells will still survive. You add AKT inhibitor, MAP kinase inhibitor, NFKB inhibitor, there you are adding too many drugs that causes toxic side effects to normal cells also. Okay. And even if you add 3 different drugs, then maybe there is a 4th mutation that may survive still. So because of this heterogeneity of mutations, one type of cancer cell has one mutation, another has another mutation, so on. It has become almost impossible to cure cancer. That is why even after chemotherapy, initially the tumor size reduces, but you will have still leftover cancer cells, like these ones which are escaping the treatment. They will again proliferate and again form the cancer. How do we now treat cancer? Do we know what to know what to target to kill cancer? Immunotherapy is one promise, but that is also not very successful. Okay. Now this is in one cancer, one patient, one cancer tissue. If you talk about different cancers, in one patient tissue cancer cells, within one patient, one tissue, you have so many different mutations. You cannot do that also. If you talk about brain cancer, breast cancer, leukemia, lymphoma, myeloma, colorectal cancer, different cancers, the mechanisms on which the cancer cells depend on survival are even more complex, more broader complexity. Can we eventually ever get a single drug that has broad spectrum anti-cancer activity? That's the question that we want to study. Is that clear? To some extent, we have multiple types of mutations. In different tumors, the mutations are even more diverse. And there is no way to kind of target each and every mutation with wide variety of drugs. Can we find single drug that can take care of all mutations? Okay? So that we develop a broad spectrum cancer. So that's our idea to begin with. So what we want to study is that despite all this heterogeneity, the mixture of mutations, we can identify a commonly operated mechanism required for the survival of cancer cells. Across all different cancers, if you can come up with one mechanism on which all the cancer cells depend for survival, we can target that mechanism and hopefully come up with a broad spectrum cancer. That's the question that's the aim, objective we started with, and we focused for that on JAK-STAT signaling pathway and NFKB signaling pathway. I will talk something about this very quickly. And this pathway, we have very nice finding, but maybe if chance comes, if I visit you again, if you are not too bored with me, and if you invite me again, then I'll talk about the others. Okay. So what is the kind of hypothesis we have? Blood cancer, ఎంత టైం ఉంది ఇంకా? Blood cancer, as you all know, blood cells becoming cancers. They swim around in the blood vessels. They are not stuck to any place. They swim around in the blood circulation, right? Whereas solid tumors, such as brain cancer, stomach cancer, or breast cancer, they form a tumor lump at one place it is attached to this place. Okay, like this. Yeah, here. So the solid tumors, such as brain cancer, breast cancer, and so on, they are attached to one place to begin with. The difference from the blood cancer is that these cells are swimming around in the bloodstream. They are not attached to one place. That's the fundamental difference. But these cells, when they grow in bigger size, when the tumor becomes really big, there is not enough room for example, here we have, let's say 200 students now. If you bring 200 more students, there is no space. You know, like that, if the tumors grow out of proportion, there is no more room for further cells to be produced. Therefore, they have to leave that space and go somewhere else in the body and form a tumor again. That is called metastasis. Okay. Now listen to me carefully. The cells, for them to move somewhere else, they have to detach from the place where they are attached, go into the blood circulation and migrate. In the blood circulation, they have to detach and enter the bloodstream and migrate to somewhere else. Now we have, you should have observed two common features in the blood cancer and the metastatic cancers which are trying to go somewhere else. Okay, this is also in the bloodstream, swimming around to find and go to a second place and attach there and form tumor. During that, between these two phases, it is also swimming in the blood. The blood cancer cells are also swimming in the blood. They must be facing same environmental stress for survival. If you are in this class, maybe you are fine. If you go to physics class, you'll be really killed out of boring subject. I mean, I don't want to say physics is bad. I'm sorry. Okay. I took a very bad example. I apologize. My brother is a physics guy. He always insulted me, so I had some kind of a personal thing there. So, I mean, let's take the other way example. Let's say that you are all physicists and you go to an immunology class taught by me. It will be really boring like hell and you have stress. Okay? So likewise, these cells which are supposed to be somewhere adherent, it's in the bloodstream. The environment is not so suitable for it. It undergoes stress. The blood cancer cells are also in the same environment. So maybe these two cells, the blood cancer cells and the metastatic cancer cells, are experiencing same type of stress and same type of pressure to survive. So a common survival mechanism might be operational in the blood cancer cells and the metastatic cancer cells. That's the question that we want to find. So what we did was, I don't want to go through all these too difficult. What we found was eventually there is a molecular complex, a signal, a gene regulatory complex that is essential for the survival of these circulating metastatic cancer cells and blood cancer cells, and that is basically a complex formed by NFKB. And just to conclude in 2-3 slides, or maybe 4 or 5 slides. So NFKB is a transcription factor. It is required for, it is absolutely essential for all normal homeostasis, means for all our immune system to function. NFKB is a must. If you do not have functional wild type normal NF-kappa B, immune system is collapsed. You will get a lot of infections. And likewise, if the NF-kB is mutated from the beginning, the development of the humans and mice will be highly defective. Okay, abnormal. So it is required for normal functioning, but it is also involved in cancer growth due to variety of mechanisms. I will not go into too many details. If anybody wants to learn more into this, you are very welcome to contact me at any point of time. I'll be very happy to interact with you guys further. Okay, so NF-kB is a transcription factor. It has different members, it's a family of transcription factors with all these members. And I'll skip this. Yeah. So this is the mechanism by which NF-kB is activated. Very quickly, when the cells are on the resting state. Okay, I said NF-kB is important for immune responses, right? Immune response will happen only when there is infection in your body. When there is no infection in your body, you don't need to activate NF-kB. Okay. In layman's example, if you are sitting in this class, you have to activate your brain to understand the concept of immunology. If you are in a physics class, you don't need to activate your brain to understand immunology. Right? Likewise, if there is no infection, you don't need NF-kappa. When infection is there, NF-kB has to be activated. It's a transcription factor. It regulates genes, so it has to go to the nucleus and bind to the DNA. Okay. If it is not in the nucleus, what is the cytoplasm, it's useless. So what happens is under the resting conditions, NFKB is retained, inhibited in the cytoplasm, but when the signal is activated, it goes to the nucleus and transfers various genes to produce immune response. But coming back to the cancer story, what we found was there's a complex called REL-A-YY1. It's a two proteins, it's a heterodimer. REL-A is one of the NFKB family members. YY1 is another transcription factor. These two form a complex with each other, means they interact with each other. And we study that interaction by various methodologies, immunoprecipitation and Western blots and so on. Okay. Yeah, so here. So the REL-A and YY1, they form a complex, okay? And their interaction is required for cancer cell survival, multiple myeloma in this blood cancer. If we remove REL-A from blood cancer cells, all of them, several different types of cell lines are dying. They are just killed if you remove REL-A. If you remove YY1, also these cells are all dead. Okay, follow me carefully, last few slides, just 5 more minutes. If you remove REL-A from blood cancer cells, they all die. If you remove YY1 from blood cancer cells, they also die. And we also find that REL-A, YY1, they bind with each other. That's another finding. And then, yeah, you can see here that the control cells, the blood cancer cells, any cancer cell, that the control cells, when we inject into the mice, they form a tumor, if you can see this, okay? Whereas the cells which are lacking REL-A or YY1, they don't form the tumor in mice, because you need REL-A and YY1 for the survival of the blood cancer cells, okay? And then what we did was, what we eventually found was that this complex, REL-A and YY1 together, need to go to the nucleus, bind to the gene promoters to provide survival to the cancer cells. This complex formation is required in myeloma. Okay. And now the question is the same. Blood cancer cells are migrating in the circulating in the blood vessels. Metastatic cancer cells also are circulating in the blood vessels. Is it possible that the REL-A YY1 complex is also found in metastatic circulating tumor cells to provide their survival? In blood cancer, we know if you remove REL-A or YY1, they die. Is this complex also found in metastatic cancer cells? The answer is yes. Yeah, this is the immunoprecipitation to show that REL-A YY1 will interact with each other in metastatic colorectal cancer cells. We found the same in breast cancer cells. And eventually, what happens is that the complex is essential for regulating survival, genes that are required for the survival of blood cancer cells and metastatic circulatory tumor cells. Okay, I leave it there. I don't want to go into too many details. I would acknowledge. I'll skip all this. Yeah, so what we found was, to summarize, in blood cancer cells like multiple myeloma, REL-A YY1 complex is formed. It goes to the nucleus and regulates some genes to provide survival to the myeloma cells. If you interfere with the function of these REL-A YY1, myeloma blood cancer cells will die. Same thing in colorectal cancer. We find this complex, REL-A YY1 complex, for the survival of colorectal cancer cells and breast cancer cells, suggesting that REL-A YY1 complex is commonly found in blood cancer cells, metastatic cancer cells, and if we can develop a drug to target this, the expectation is that we have a broad spectrum cancer drug to kill blood cancer cells and metastatic cancers. That's the idea. Okay. Yeah. So most of this work was done by my student Rama Devi, and the other pathway, JAK-STAT pathway, I did not explain. It's much more complex mechanism actually. And then some of the computational studies were done by Sahil Sarogalla, Shreya Mudherkar, Asmita Das, Maina. These are all my students. My collaborators, one of my guru, Michael Karin, is also my collaborator. He provides any reagent that I ask. And Professor Lee Blacksago from Mayo Clinic, Arizona, Professor Marta Chesi also from Mayo Clinic, Arizona. Our research is funded largely has been funded by Welcome Trust DBT India Alliance and SELF DST, ANRF. That's my phone number if anybody wants to contact me in future. The most beautiful pictures you can actually keep in your minds for several decades if you are interested in biology eventually. Michael Karim, okay? He is my guru. Yeah, he was the one who gave me the title that I am not useless. Okay, a person with 350,000 citations giving me the title that I am not a useless fellow, I take it as the biggest compliment in my life and take it further. Okay, and the other side of Michael, okay, he's also very fun-loving guy. You can see his pose here and all that stuff. He's very angry in the lab. If you don't work hard and if you do mistakes, he will be really angry at you. He will kill you. He may even ask you to leave the lab and go and all that stuff. So that's about him. Okay. I thank all of you for your kind attention and we'll be happy to take any questions.
Any questions from the students? Try to ask one or two.
But they are interested at least some aspects.
Any faculty? Faculty the students.
Because when they are developing in the body, they are educated in such a way that you react only to foreign invaders into the body, not to your own body cells. In layman's terms, I tell you, you recruit security guards at the gate, right? The security guard is educated to recognize who belongs to this college and not to obstruct them from entering into the college. He should also identify who is foreign to this college, who is not belonging to this college. Should stop that person, and if anybody is invading, should fight them off. Immune system is also same. It is there to safeguard you from invaders into your body, educated in such a way, and it is also educated to recognize what belongs to your own body. During this education process, if any errors happens, autoimmunity happens. They fail to recognize your cells as your own cells and recognize them as foreign and start reacting to your own cells and create havoc in the body. That's autoimmunity in a nutshell. There are a lot of mechanisms on why they fail, how they fail, and how they are educated to recognize self and foreign and all. Maybe next time. Or anybody can reach out to me. That's fundamental aspects I teach in immunology class. Yeah, sure.
Thank you. I would like to thank him for referring to me as a good student to make him more bad, effective way of presenting him as a bad student, right? But I would like to mention one incident. He was telling that IAC is the top destination. I completely agree. Being a biochemistry background, being a biochemistry, so I appeared after my MSc. I appeared IAC for PhD position. There was to be 2 interviews. In the first interview, I cleared, and one of the committee member and came to me and congratulated me after the interview. But second interview, I failed. I could not clear it. But he was successful.
After getting proper number of...
okay, he has only taken some more time than me, right? He has only taken a little more time than me.
No, it took long time.
Yeah.
After getting Muhammad number of times of attempts, I cleared. But he got a job in Park. That's why he did not try again.
So it is like that. But we have to find what I wanted to say. And then why was that? Because I also felt in the second interview, because my graduation was in Telugu medium at that time. BSc Telugu medium lo chadiva. So some of the terminology, the tadpole retrogressive metamorphosis, that was the question I was asked to me, Thyrox syndrome. So I said medium, Telugu medium, I am not able to answer that. But Ramaswamy was referring to me, he said, no, you can take Telugu, you can explain me in Telugu. But somehow when I was not, I lost hope in the mid of the interview. So things will be like that, but we have to face it. So it is not that if ISC will have our own, right? I have BS, right? So it's not that only one, but for him, ISC was the only destination. Okay.
Yeah, it was to kind of give a answer to my brother. It's not for my own gain. So anyway, in the meantime, do you have a sentence? Stop. Yeah, I'll quickly say a couple of words. In the meantime, we have another fantastic scientist here, Professor Naresh Sepuri from University of Hyderabad. He just recently published in Nature Communications. Publishing in Nature Communication from India is a fantastic experience. Okay, and he is very good science. Okay, STAT3 signaling pathway in mitochondrial functioning. Hopefully tomorrow I will talk about science. Please don't miss the opportunity to listen to him. Fantastic work, actually. That's one thing. And last but not least, I'll tell you one last sentence and then I'll stop here. When I went to Michael Cunning's lab, I was doing routine stuff. Routine stuff. That means somebody found something. You also add a little bit more to that, saying that I'm also doing some things. Okay. So then I was doing very simple and ordinary things. He was very angry and he told me one thing to remember for life. Actually, two things. Never do things like I am also doing it. Somebody is doing, I am also doing it. So you are not unique. You are not so what? It's not to somebody else. You have to do things only I can do this. To contribute uniquely to whatever field you work, whatever you do in life, at least do one thing that is unique to you in your life. So that makes a big contribution to whatever field you are. That's one thing, okay? So remember that.